In the quiet moments when the day begins to exhale and the mind starts to drift, it can be soothing to imagine the gentle science that sustains you from within. Nutrition, though often thought of simply as eating, is in truth a long and peaceful story, one told through molecules, enzymes, and the steady rhythm of energy moving through the body. Each time you take in food, you are offering your cells a new collection of tiny structures built by plants, animals, sunlight, and soil. These structures come with their own histories, their own patterns of atoms arranged in ways that nature has refined over millions of years. And as they enter the body, they begin a slow, intricate transformation, becoming part of the warmth that surrounds you, the strength that supports you, and the quiet motion that continues even as you rest. This gentle conversion of food into life-sustaining energy unfolds through biochemistry, a field that reveals the hidden harmony between nourishment and cellular function. At its heart, biochemistry shows that life is built upon constant movement, tiny reactions happening in every corner of the body, each one contributing something essential. These reactions are not loud or sudden. Instead, they flow softly, like ripples spreading across still water. Molecules drift toward one another, bonds loosen in response to subtle cues, and enzymes guide each process at a pace that feels almost meditative. In this slow sequence of events, the foods you eat are gradually reshaped into forms your cells can recognize and use. Carbohydrates, fats, and proteins, three familiar categories from the dinner plate, carry different kinds of potential when they enter the body. Carbohydrates bring quick, accessible energy. Fats offer long-lasting reserves. Proteins provide structure, repair, and many of the tools the body uses to maintain itself. Yet before these nutrients can offer anything meaningful, they must pass through a series of calm transitions. Starches are eased into simple sugars, fats are separated into fatty acids, and proteins are untangled into their amino acid components. These gentle decompositions occur through the quiet work of enzymes and fluids released along the digestive tract. The process is neither rushed nor forceful. Instead, each stage prepares the next, creating a smooth flow toward absorption. What is remarkable is how familiar this entire process is to the body. It has performed these transformations since long before memory, carrying forward a wisdom shaped by countless generations. Every enzyme knows its role. Every cell recognizes the signals that accompany digestion, and every organ collaborates in the wider task of turning food into fuel. The digestive system acts not as a simple passageway but as an orchestra, where each section contributes to the soft unfolding of biochemical changes. Even when the mind is quiet or you've drifted into sleep, the body continues its work, gently breaking down nutrients and sending them where they need to go. Once nutrients are released from food, they begin their journey through the bloodstream. This movement is slow and purposeful. Glucose derived from carbohydrates may be carried to a muscle cell preparing for a light contraction, or to a neuron needing energy to maintain a quiet electrical gradient. Fatty acids might travel to the liver, where they are slowly shaped into ketone bodies during times of fasting or quiet metabolic shifts. Amino acids may be delivered to tissues where repairs are needed or where new proteins are waiting to be assembled. Each molecule finds its place through the calm guidance of hormonal signals and concentration gradients, mechanisms that keep the body in steady balance. The chemistry that unfolds inside each cell is even more subtle and perhaps even more soothing to imagine. Within tiny compartments like mitochondria, reactions take place with extraordinary precision. Glucose is gradually eased through pathways that release energy step by step, not all at once. This slow release protects the cell, ensuring that each bit of energy can be used efficiently. The molecule ATP, often called the energy currency of the cell, is produced in small bursts that fuel gentle processes, moving ions across membranes, building proteins, and maintaining the quiet hum of cellular life. Even at rest, cells rely on a steady supply of ATP, and the body continues to make it through pathways that have remained unchanged across the history of living organisms. Reflecting on this chemistry can bring a sense of calm, because it reminds us that much of what sustains life happens without effort or thought. The body tends to its needs with a kind of internal grace. It digests food without instruction, absorbs nutrients without hesitation, and distributes energy in the ways that best support balance. Excess glucose may be tucked away gently in the liver or in muscle tissue as glycogen, saved for a later time when the body might need a little extra support. Fats are placed in adipose tissue, waiting patiently to be called upon. Proteins circulate in pools that can be drawn from when growth or repair is required. Nothing is wasted, and nothing moves without purpose. The interconnectedness of these biochemical processes is also comforting. Carbohydrates can become fats, fats can become ketone bodies, and amino acids can enter pathways that once belonged only to sugars. The body adjusts these transitions according to what is available and what is needed, maintaining a sense of internal harmony. Hormones whisper instructions. Insulin encourages cells to take in glucose. Glucagon prompts the release of stored energy. Cortisol adjusts the flow of fuels during stress or fasting. These hormonal signals are steady and nuanced, guiding metabolism in ways that preserve the body's equilibrium. This peaceful introduction sets the tone for the deeper exploration to come. As you move into the next parts of this journey, you will follow nutrients more closely, observing how they travel through digestion, how they slip into the bloodstream, and how they are welcomed into the inner spaces of the cell. But for now, the breath you take, the warmth you feel, the calm settling around you, all of it is shaped by the slow and steady transformation of nourishment into energy and form. This soft foundation will carry you into the next chapter, where the digestive system begins its quiet work. 2. The digestive system as a peaceful pathway. The journey of nourishment truly begins the moment food enters the body. Yet this beginning is not abrupt or hurried. Instead, it unfolds in a gradual, rhythmic sequence that mirrors the calm order of nature itself. The digestive system, often imagined as a busy corridor of activity, can also be understood as a gentle, continuous pathway where food is welcomed, softened, and slowly transformed into forms the body can understand. As you ease into this exploration, let the pace become slow and soothing. Much like the movements of the digestive organs themselves, think of this system not as a place of harsh breakdown, but as a long, peaceful landscape where each region contributes quietly to the unfolding story of nourishment. It all starts with the simple act of taking in food, a gesture so familiar that it often passes unnoticed. Yet at this very first moment, the body begins preparing itself, releasing moisture into the mouth and awakening sensory pathways that help anticipate what is to come. The mouth is the first gentle chamber of digestion. Teeth do not forcefully crush food. Instead, they guide it into smaller pieces, making it easier for the rest of the pathway to work at its natural pace. Each movement of the jaw is steady and predictable, a mechanical beginning that pairs perfectly with the chemical changes happening alongside it. Saliva, produced effortlessly, contains enzymes that begin the early stages of breaking down carbohydrates. These enzymes, such as amylase, do not rush the process. They simply start the slow unraveling of long starch molecules into smaller sugars, marking the first quiet step toward nourishment. Once the food has become soft enough to move on, it is shaped into a bolus and gently guided to the back of the throat. The swallowing process is often described in textbooks as a complex series of muscular contractions. But in the lived experience of the body, it feels smooth and unintrusive. The bolus slips past the pharynx and enters the esophagus, a long tube whose sole purpose is to carry it downward. Muscles along the esophagus engage in a rhythmic pattern known as peristalsis. This pattern does not force the food downward with pressure. Rather, it nudges it along, wave after wave, in a motion that feels almost tidal. It is a serene progression, one that continues faithfully whether the body is awake or resting. The stomach is often imagined as a place of strong acids and intense churning, yet the reality is subtler and more balanced. Within its curved walls, food is greeted by gastric juices that begin dissolving proteins and softening the food further. The acidity here does not seek to harm. Instead, it creates the ideal environment for enzymes to do their work, unfolding proteins into simpler structures that the body can later reshape as needed. The muscles of the stomach do not rush the process either. They make slow, sweeping motions, swirling and mixing the food with just enough force to encourage further breakdown. Over time, the bolus becomes a smooth mixture called chyme, which is easier for the intestines to handle. The stomach releases chyme into the small intestine in measured amounts, never overwhelming the process. The next portion of the pathway. This slow release reflects the natural rhythm of digestion, one that supports both efficiency and comfort. As chyme enters the small intestine, it encounters a new blend of secretions, alkaline fluids from the pancreas, and bile from the liver and gallbladder. These secretions serve as gentle helpers, neutralizing the stomach's acidity and preparing fats, carbohydrates, and proteins for deeper digestion. Pancreatic enzymes continue the work that began in the mouth and stomach, while bile emulsifies fats, turning them into droplets that enzymes can reach more easily. Even this emulsification is gentle in nature. It does not force fats apart, but rather encourages them to disperse, making digestion more harmonious. The small intestine is long, winding, and deeply purposeful. Its inner surface is lined with folds, villi, and microvilli, structures that increase the surface area available for absorption. These structures do not grasp nutrients aggressively. Instead, they provide a broad, welcoming landscape where molecules can slip across the intestinal wall at their own pace. Carbohydrates become simple sugars, proteins become amino acids, and fats become fatty acids and monoglycerides. As these molecules are released, they drift into the spaces between cells, guided by chemical gradients and transport proteins that know precisely when to open and when to close. Each nutrient finds its way into the bloodstream or the lymphatic system, beginning a new chapter in its quiet journey through the body. Even as nutrients are absorbed, the small intestine maintains a gentle circulation of movement. Its muscles contract softly in a pattern called segmentation, which helps mix the chyme and ensure that all nutrients are exposed to the intestinal lining. This mixing is far from harsh. It is a slow and even rhythm, almost like the swaying of grasses in a light breeze. Through this continual motion, digestion and absorption proceed with both grace and efficiency. The body never rushes these steps, because nourishment is not something to be hurried. It is something to be received and transformed with care. What remains after absorption moves into the large intestine, a space devoted not to digestion but to reclamation. Here, water is drawn back into the body, along with minerals and electrolytes that are needed for balance. The movement through the large intestine is even slower than through the rest of the digestive tract, reflecting its quieter purpose. Beneficial bacteria that live within this space help break down remaining fibers, producing compounds that the body can still use. These bacterial communities thrive in harmony with the body. Their work is unhurried, and their presence adds another layer of calm cooperation to the digestive process. As the final remnants of food pass through the last parts of the large intestine, the body prepares to release what is no longer needed. This release, too, is a natural and unforced part of the digestive cycle. It completes a journey that may have taken many hours, yet never felt rushed. Every step of digestion, from the first bite to the final release, has been guided by gentle movements, chemical balances, and a quiet orchestration of organs working together. It is a reminder that the body functions in a state of calm coordination, trusting its own rhythms to carry out essential tasks. Reflecting on the digestive system in this way can change the way you experience nourishment. Instead of imagining digestion as a process that happens out of sight and out of mind, you might begin to see it as a flowing sequence of supportive actions, each one designed to help you thrive. Every wave of peristalsis, every drop of enzyme, every moment of absorption contributes to the steady supply of energy that keeps your body alive. This peaceful pathway echoes the deeper truth that life is sustained by processes that unfold quietly, faithfully, and without need for attention. As you prepare to continue into the next part, you may find comfort in recognizing how much care the body takes with every meal. The digestive system sets the stage for all that follows, laying down the foundation for energy production, cellular repair, and metabolic balance. With this foundation in place, the next chapter will explore the enzymes that catalyze much of this transformation, the silent workers that guide molecules toward their next gentle step. 3. Enzymes, silent catalysts that begin the transformation. In the quiet depths of the body, away from conscious awareness, and far from the noise of the external world, there exists a family of molecules that carry out their work with extraordinary grace. These molecules are enzymes, the gentle catalysts that allow the transformation of food to unfold in a rhythm that sustains life. Without them, digestion would be slow to the point of impossibility, metabolism would stall, and the energy locked within every bite of food would remain inaccessible. Yet enzymes do not demand recognition for their efforts. They do not draw attention to themselves. Instead, they drift through their environments with quiet purpose, guiding reactions along the paths nature designed, ensuring that every nutrient finds its way to its next stage with softness and precision. Enzymes begin their work even before the food has fully entered the body. As soon as the aroma of a meal reaches the senses, the body anticipates nourishment, and saliva begins to flow. Within this clear liquid, the first digestive enzyme, salivary amylase, waits patiently. It slips between the long chains of starch molecules, encouraging them to unravel into smaller fragments. This process is not forceful. It is more like the gentle loosening of a knot. As food moves through the mouth, mixed with saliva through slow and mindful chewing, area, giving them space to work at a comfortable pace. They do not break molecules through sheer strength. Instead, they lower the energy needed for bonds to shift, helping reactions unfold almost naturally, as though the food were meant to become nourishment all along. When the food moves into the stomach, it enters an environment defined by warmth, moisture, and carefully measured acidity. Here, new enzymes begin their part of the journey. Gastric lipase starts breaking down fats, while pepsin begins unraveling the tightly folded structures of proteins. For a moment, it may seem surprising that these transformations happen in such an acidic space, but the enzymes that reside here are adapted to work best under these conditions. They are shaped in ways that allow them to remain steady and functional even when surrounded by powerful gastric acids. Their actions remain controlled, slow, and purposeful. They take the proteins and fats that once gave structure and energy to living organisms and guide them toward simpler components, making them ready for the next stage of digestion. Even as enzymes work in the stomach, they are supported by a system that understands the importance of balance. The stomach does not simply release acid and enzymes randomly. It adjusts the timing and quantity based on the presence of food, the type of nutrients being digested, and feedback signals from the small intestine. This ensures that enzymes never become overwhelming and that the environment remains just right for their gentle work. The body treats enzymes with the same care it offers to all essential processes, maintaining conditions that allow them to operate smoothly, steadily, and with remarkable efficiency. When the partially digested food moves into the small intestine, the world of enzymes expands significantly. The pancreas, an organ tucked quietly behind the stomach, plays one of the most important roles in this stage. It releases a mixture of enzymes into the small intestine, each tailored to a specific type of nutrient. Pancreatic amylase continues the work of breaking down carbohydrates. Proteases such as trypsin and chymotrypsin further dismantle proteins into smaller peptides and eventually into individual amino acids. Lipases turn fat droplets into absorbable components. These enzymes function like a gentle team, each one designed to complement the others, each one responding to the presence of certain molecules, and each one working at a pace that aligns with the body's needs. The release of pancreatic enzymes is carefully regulated. The moment acidic chyme from the stomach enters the small intestine, the lining of the intestine releases hormones that signal the pancreas to send its enzymes forward. At the same time, bile from the liver is released to emulsify fats, making them more accessible to lipases. Nothing in this process is rushed. It is a series of responses, each triggered by the step before it, forming a chain of gentle guidance. Enzymes do not act alone. They are part of a broader conversation among organs, tissues, and chemical messengers, each contributing to the quiet breakdown of food into its simplest forms. As these enzymes move along the small intestine, they encounter the brush border, the finely textured surface of the intestinal wall, covered with microscopic projections called microvilli. This surface hosts its own family of enzymes, anchored in place and waiting for the nutrients that pass by. These brush border enzymes complete the breakdown of carbohydrates into simple sugars and peptides into individual amino acids. Their work is subtle and elegant, occurring at the final moment before absorption. They do not travel with the flow of chyme. Instead, they wait patiently, transforming nutrients only when they draw near. In this way, digestion becomes not a single event, but a series of gentle transitions, each one guided by the presence of enzymes adapted perfectly to their environment. What is truly remarkable about enzymes is how specific they are in their actions. An enzyme that breaks down carbohydrates will not break down proteins. One that assists with fats will not interact with starches. This specificity arises from the shapes of enzymes, each one formed in a way that allows only certain molecules to fit against its active site. The interaction is often compared to a lock and key, though in truth it is more fluid than that. The enzyme subtly shifts its shape to embrace the molecule it acts upon, forming a momentary connection that allows the reaction to proceed. Once the transformation is complete, the enzyme releases the product and returns to its original form, free to assist again. Through this repeated cycle, a single enzyme molecule can help thousands of reactions unfold, all without being consumed or altered permanently. Enzymes also reflect the delicate sensitivity of the body. They work best within narrow ranges of temperature and pH, and the body maintains these conditions with great care. A slight increase in stomach acidity triggers a shift in enzyme activity. A rise in temperature accelerates reactions before the body gently cools itself again. Even small changes in water balance can influence how enzymes function, which is one reason hydration plays such a quiet but important role in digestion and metabolism. Every aspect of the internal environment exists in harmony with enzymes, forming a system that supports life with quiet consistency. Beyond digestion, enzymes continue their gentle work inside cells, guiding metabolic pathways that produce energy, create new molecules, and maintain homeostasis. Just as digestive enzymes help break nutrients down, cellular enzymes help rebuild them into the structures and fuels needed for daily life. Glycolysis depends on a sequence of enzymes that shepherd glucose through 10 small steps. The citric acid cycle requires enzymes that carefully coordinate reactions within the mitochondria. Even the production of ATP, the energy currency of the cell, relies on enzymes embedded in the inner mitochondrial membrane. These enzymes never stop working, even as the body sleeps. They keep the flame of metabolism burning softly, ensuring that cells receive the energy they need without interruption. Enzymes are, in many ways, the guardians of transformation. They act not through force, but through guidance. They do not break anything. They simply help things become what they are meant to be. Without them, food would remain unchanged, energy would remain locked away, and the body would lose the gentle momentum that keeps it alive. By acting softly and steadily, enzymes reveal a truth that is easy to forget in the fast pace of daily life: that much of what sustains us is quiet, subtle, and almost invisible. As we move into the next part of this journey, we will follow one of the major nutrient groups more closely: carbohydrates, the body's primary source of quick and immediate energy. But before stepping forward, it is worth pausing for a moment to appreciate the elegance of enzymes, the silent catalysts that make all nutrition possible. 4. Carbohydrates, nature's readily accessible energy sources. Carbohydrates move through the world with a simplicity that belies their profound importance to life. They are found in the gentle tissues of fruits, the sturdy grains that have supported civilizations, the roots that grow quietly beneath the soil, and the leaves that stretch toward the sun. They are, in essence, molecules born from light. Through the patient work of photosynthesis, plants take sunlight, water, and carbon dioxide and use them to build carbohydrates, storing the sun's energy in the form of chemical bonds. When we eat these plant foods, or animals that have fed on them, we tap directly into that stored energy. It becomes the warmth that surrounds us, the fuel that lets us walk, think, breathe, or drift to sleep. Many organisms, the most immediate and comforting source of energy. At their core, carbohydrates are composed of carbon, hydrogen, and oxygen arranged into rings or chains that vary in length and complexity. Some are as small as a single sugar unit. Others are long, branching structures that take time and specific enzymes to unravel. Yet no matter their shape or size, carbohydrates ultimately become a molecule that cells recognize effortlessly: glucose. Glucose is the quiet cornerstone of energy metabolism, a molecule that the body can break down quickly when needed or store gently for later use. Before glucose can become useful, however, it must be released from the food that carries it. This release is part of the digestive story, part of the slow and peaceful unfolding of nutrients that begins long before glucose reaches the cell. When carbohydrates enter the body, their journey begins in the mouth, where the enzyme salivary amylase starts loosening the bonds of starch. But this first step is just a whisper of the work to come. Many carbohydrates pass through the mouth only partially digested, and as they travel down the esophagus and into the stomach, the acidic environment temporarily pauses their breakdown. Carbohydrates wait there quietly, held in the warm, churning mixture until the stomach releases its contents into the small intestine. It is in this next stretch of the digestive pathway that the transformation unfolds more completely. The small intestine becomes a place of careful attention, where enzymes from the pancreas and the brush border lining work together to convert complex carbohydrates into smaller and smaller components. Pancreatic amylase acts upon long chains of starch, cutting them into disaccharides and shorter fragments. On the surface of the intestinal lining, tiny enzymes such as maltase, sucrase, and lactase take on the final steps, splitting these disaccharides into individual monosaccharides: glucose, fructose, and galactose. These simple sugars are small enough to be transported across the intestinal wall, slipping gently into the bloodstream where they can begin their roles in metabolism. Among these sugars, glucose takes center stage. It is the form preferred by the brain, which relies heavily on a steady supply of it to maintain its quiet electrical patterns and complex networks of thought. Muscles also draw upon glucose, especially during periods of activity, whether movement is calm and slow or brisk and demanding. Even the red blood cells, which lack mitochondria, depend entirely on glucose for their energy needs. Because of this, the body has developed a delicate system for ensuring that glucose remains within a narrow range, never too high, never too low, allowing cells to function smoothly. Once glucose enters the bloodstream, it begins to influence the body's internal balance. The presence of glucose stimulates the pancreas to release insulin, a gentle hormone that acts like a guide. Insulin signals cells to open their doors, allowing glucose to enter. Some of it will be used immediately, burned at a slow and steady rate to maintain the energy needs of the cell. Some will be stored as glycogen in the liver or muscle tissue, a compact and efficient form of reserve energy. And some may even be converted into fat when reserves grow full, saved for long-term needs. All of these shifts occur quietly, without sensation, yet they shape the body's level of energy and comfort. The speed at which carbohydrates release their glucose varies, and this variety mirrors the diversity of foods themselves. Simple sugars found in fruits, milk, and honey enter the bloodstream more quickly than the long chains found in whole grains or legumes. But this difference is not something the body treats with urgency. It responds with the same careful adjustments of hormones and transporters, maintaining equilibrium with a rhythm that feels almost soothing. Even the act of storing glucose in the product liver as glycogen can be imagined as a form of gentle preparation, setting aside energy for the hours between meals or for moments when the body needs a quiet boost. For many people, carbohydrates bring not only biochemical nourishment but also emotional comfort. The foods rich in carbohydrates, bread, rice, potatoes, pasta, fruits often carry cultural significance and memories of meals shared with loved ones. While these experiences are separate from the chemistry itself, they echo the body's natural connection to this source of energy. Carbohydrates are, in many ways, familiar companions. They enter the bloodstream with a certain predictability, offering a sense of steadiness that aligns with the body's natural rhythms. Not all carbohydrates behave in the same way within the digestive system, and understanding this can ease any concerns about their diversity. Fiber, for example, is a type of carbohydrate that the body cannot fully digest. Yet, it plays an important role by slowing the movement of food through the digestive tract and supporting the health of gut bacteria. These fibers gently influence how quickly sugars enter the bloodstream, preventing abrupt changes in energy levels. They also add volume to food, supporting digestive comfort and regularity. Even though fiber does not become glucose, its presence shapes the overall experience of digestion in subtle and beneficial ways. The body's relationship with carbohydrates is deeply intertwined with the flow of metabolism. When glucose enters a cell, it is welcomed into a pathway known as glycolysis. This pathway is the first step in the gradual release of energy stored within the molecule. Glycolysis splits glucose into two smaller molecules, releasing a small amount of ATP, the energy currency of the cell. This gentle release supports everything from maintaining cell membranes to powering quiet cellular movements. If oxygen is readily available, these smaller molecules enter the mitochondria, where the citric acid cycle and electron transport chain generate more ATP with slow efficiency. Through this process, carbohydrate-derived energy becomes warmth, motion, and life. Even when glucose levels drop between meals, the body remains calm. The liver slowly releases stored glucose from glycogen, maintaining balance. Hormones work quietly to ensure that glucose remains available to the brain and other essential organs. If the hours pass and glucose stores become low, the body transitions smoothly into alternative fuels such as fatty acids or ketone bodies. These shifts occur without abrupt changes, maintaining a sense of metabolic peace. Carbohydrates, despite their varying forms and sources, share one central purpose: to provide the body with energy that is easy to access, easy to use, and deeply connected to the rhythms of life. They move through the digestive system with grace, are absorbed with care, and enter the pathways of metabolism with quiet confidence. Transformations fuel the brain's thoughts, the heart's steady beat, and the muscles' gentle contractions, all without demanding attention. As we move into the next part of this calm journey, we will follow carbohydrates more closely into the bloodstream, exploring how glucose travels through the body and how it becomes welcomed into the cell. But for now, it is enough to recognize carbohydrates as the sunlight-derived companions they are, bringing warmth and energy into life through the simplest and most ancient biochemical pathways. 5. Carbohydrate digestion from mouth to intestine. Carbohydrate digestion is one of the most fluid and graceful sequences within the body, beginning the moment food touches the lips and continuing in a slow, uninterrupted flow until the nutrients reach the bloodstream. This journey is gentle, unfolding in deliberate steps that mirror the steady rhythms of nature. Carbohydrates, whether they come from grains, fruits, roots, or vegetables, carry with them stored sunlight and energy captured by plants. As they enter the body, they release this energy in a measured, comforting way, allowing the system to draw from it at just the right pace. Understanding the path of carbohydrate digestion from mouth to intestine is like watching a quiet story unfold, one where each chapter leads calmly into the next. When food is first brought into the mouth, the body recognizes the anticipation of nourishment, and a soft wave of saliva begins to form. This saliva contains water, minerals, and most importantly, the enzyme salivary amylase, though the mouth is not where most carbohydrate digestion takes place. The significance of this early encounter is profound. Amylase begins separating long starch molecules into shorter fragments, gently loosening the bonds that hold them together. The movements of chewing create more surface area, making it easier for the enzyme to begin its work. Break into maltose and smaller sugars, though many remain unchanged. The mouth's environment is mild and neutral, creating a comfortable starting point for the long journey ahead. Swallowing moves the food into the esophagus, where rhythmic muscular waves called peristalsis carry it downward. During this quiet descent, carbohydrate digestion pauses. The esophagus is a passageway, not a place for chemical transformation, and it transports the food with calm efficiency. Once the bolus reaches the stomach, it enters a new environment. The stomach is warm, acidic, and designed primarily to begin protein digestion and prepare all food for further breakdown. Here, the acidity temporarily halts the activity of salivary amylase. Carbohydrates rest within the churning mixture of gastric juices and partially digested proteins and fats, protected from any harsh or abrupt changes. The stomach does not rush them onward. It holds the mixture until it reaches the right texture and consistency, slowly turning it into chyme, a smooth, semi-liquid form that can pass safely into the next stage. Carbohydrates spend this period in the stomach mostly unchanged, waiting for the environment in which their true breakdown will occur. When the stomach decides the chyme is ready, it releases it gradually into the small intestine through the pyloric sphincter. This slow release is essential. It ensures that the small intestine is never overwhelmed and that each portion of chyme receives careful enzymatic attention. As soon as the chyme enters the first region of the small intestine, the duodenum, it is met with a wave of alkaline secretions from the pancreas. Among these secretions is pancreatic amylase, an enzyme far more active in this environment than its salivary counterpart. The acidity from the stomach is neutralized almost immediately, creating a gentle, slightly alkaline environment where amylase can work effectively. Pancreatic amylase continues the job that began in the mouth, breaking down starches into disaccharides and smaller polysaccharides. The enzyme moves with ease through the chyme, finding long carbohydrate chains and encouraging them to separate into shorter, more manageable fragments. These fragments may include maltose, maltotriose, and various dextrins. Though the process is highly efficient, it is not rushed. The chyme moves slowly along the intestinal wall, allowing enzymes ample time to interact with it. The small intestine's smooth muscles contract in soft, segmenting motions, keeping the chyme in contact with the lining. This gentle movement feels almost wave-like, guiding the nutrients in calm circles that maximize digestion and absorption. As carbohydrate digestion progresses, the chyme reaches the brush border, a complex surface made of tiny projections called microvilli. This is where some of the most delicate and essential transformations take place. Embedded within the brush border are specialized enzymes, each one finely tuned, to break down specific disaccharides into monosaccharides. Maltase splits maltose into two glucose molecules. Sucrase divides sucrose into glucose and fructose. Lactase separates lactose into glucose and galactose. These enzymes act with remarkable precision. They do not float freely like pancreatic enzymes. Instead, they remain anchored to the intestinal lining, waiting patiently for the carbohydrates to come to them. When a disaccharide approaches, the enzyme embraces it briefly, performs its task, and releases the resulting simple sugars. These monosaccharides, glucose, fructose, and galactose, are the smallest units of carbohydrates, and their journey next turns toward absorption. The cells of the intestinal lining, called enterocytes, guide them inward through a combination of transport proteins and gradients. Glucose and galactose are absorbed actively, using sodium-dependent transporters that gently pull the sugars into the cell. Fructose enters more passively, slipping through a different transporter that allows it to diffuse at its own pace. Once inside the enterocyte, the monosaccharides move to the other side of the cell and enter the bloodstream, carried away through tiny capillaries within each villus. The bloodstream becomes the new river on which carbohydrate energy travels. Glucose enters the portal vein and moves toward the liver, the body's metabolic guardian. The liver decides how much glucose should be released into general circulation and how much should be stored as glycogen. These decisions are made quietly, influenced by insulin and other hormonal signals. The release of glucose into the bloodstream is not abrupt or uncontrolled. It occurs gradually, ensuring that the brain, muscles, and other tissues receive a steady supply of energy. Meanwhile, the small intestine continues its peaceful movements. As digestion and absorption proceed, what remains of the chyme becomes less rich in nutrients and moves toward the large intestine. Any carbohydrates that escape digestion, such as certain fibers, continue on this path. These bacteria break down some fibers through fermentation, producing short-chain fatty acids that the body can still use as energy. This final step reflects the body's thoughtful efficiency, allowing even the parts of carbohydrates that are not digestible by human enzymes to become part of the broader picture of nourishment. Carbohydrate digestion, from the first moment of chewing to the last moment of absorption, is a series of gentle shifts guided by the body's innate wisdom. Each enzyme plays its part. Each section of the digestive tract contributes a unique environment tailored to the molecules passing through it. Nothing is hurried, and nothing is wasted. The body receives the energy it needs through a process that is as patient as it is precise. As we move forward, the next chapter will follow glucose, as it travels through the bloodstream and enters the cells, where it becomes the foundation of cellular energy. But for now, the quiet unfolding of carbohydrate digestion offers a sense of comfort, a reminder that the body handles nourishment with care, purpose, and the soft pulse of natural rhythm. 6. Glucose absorption and gentle bloodstream transport. When glucose finally enters the bloodstream, it has already traveled a long and delicate path through the digestive system. From its beginnings in plants, where sunlight and water gave rise to the sugars stored within leaves, grains, and fruits, glucose now becomes a quiet participant in the body's inner harmony. Its arrival in the bloodstream is not abrupt or overwhelming. Instead, it flows gradually from the cells lining the small intestine, slipping softly into tiny capillaries that lead toward the hepatic portal vein. This transition is one of the most peaceful and essential steps in human physiology. For glucose is the body's most readily accessible form of energy, a molecule that nearly every cell can welcome and use. Once inside the blood, glucose begins its journey toward the liver, an organ that serves as the first point of decision-making. The liver receives nutrient-rich blood directly from the small intestine allowing it to sense the amount of glucose present and adjust its responses accordingly. If glucose levels are high, as they often are after a meal, the liver absorbs a portion of it and converts it into glycogen, a soft, compact form of stored energy. This storage process is like placing extra warmth in a safe reserve, waiting quietly for moments when the body might need it later. If glucose levels are moderate or low, the liver allows more of it to pass onward into the main circulation, providing energy to tissues throughout the body. These decisions are made without any conscious guidance, relying instead on hormonal signals and natural biochemical rhythms. As glucose leaves the liver and enters the wider bloodstream, it starts to circulate through the body in gentle currents. The blood vessels carry it to every tissue, from the smallest muscles of the fingers to the deep structures of the brain. Each cell along the way has the potential to draw from this shared supply. The presence of glucose in the blood triggers the release of insulin from the pancreas, a hormone that acts like a quiet messenger. Insulin does not push glucose forcibly into cells. Rather, it encourages cells to open their doors, increasing the number of transporters on their surfaces. These transporters, often called GLUT proteins, allow glucose to drift inward at a pace that matches the cell's needs. The relationship between insulin and glucose is one of cooperation and trust. When glucose levels rise, the pancreas senses the shift and responds by releasing just the right amount of insulin to help cells absorb the sugar. When levels fall, insulin release diminishes, and the body transitions to other energy sources. This interplay creates a sense of stability within the internal environment. It prevents sudden spikes or drops in glucose levels, ensuring that the brain, one of the organs most reliant on a steady supply of glucose, remains nourished at all times. Even during sleep, when the body's energy needs are lower, the liver quietly releases small amounts of glucose from its glycogen stores, maintaining a gentle baseline. For the cells that receive glucose, this molecule becomes a welcoming source of fuel. Once inside a cell, glucose can be used immediately or stored temporarily depending on the cell's purpose. In muscle cells, glucose may be converted to glycogen to support future contractions. In adipose tissue, glucose contributes to the formation of triglycerides, becoming part of the body's long-term energy reserves. In neurons, glucose is quickly channeled into metabolic pathways that maintain electrical gradients, allowing thought, memory, and perception to continue without interruption. Even the quiet work of organ function, breathing, circulating blood, filtering waste, and repairing tissues depends on the dependable support of glucose. The transport of glucose into cells is not a single-step process, but rather a soft cascade of biochemical events. When insulin binds to its receptors on the cell surface, the cell responds by recruiting specialized proteins that move to the membrane. These proteins form channels through which glucose can enter. Once glucose crosses the membrane, it encounters enzymes that begin the early steps of metabolism, such as phosphorylation, a gentle modification that keeps the molecule inside the cell and prepares it for further stages of energy extraction. Each of these steps is carefully regulated, forming a sequence that is both efficient and soothing in its predictability. What makes glucose transportation remarkable is how seamlessly it integrates with the body's broader sense of balance. After a meal, when glucose is abundant, insulin ensures that tissues store or use the sugar, preventing excess from accumulating in the bloodstream. Between meals, when glucose levels begin to fall, another pancreatic hormone, glucagon, encourages the liver to release glucose gradually. During long periods without food, the body gently transitions to using stored fats and, when necessary, produces glucose from amino acids through a process known as gluconeogenesis. These shifting patterns occur in soft waves, responding to the body's ever-changing needs while protecting the delicate equilibrium that supports life. The bloodstream acts as a quiet highway for glucose, guiding it to all parts of the body at a steady pace. The capillaries, the smallest vessels, play an especially important role, allowing glucose to diffuse into tissues where it is needed. These vessels are lined with thin, permeable walls that allow nutrients to pass through without resistance. The flow of blood remains smooth and continuous, driven by the steady beat of the heart, distributing glucose in a way that feels almost rhythmic. Even in distant regions of the body, small muscles in the hands, tissues at the tips of the toes, or cells deep in the spinal cord, glucose arrives faithfully, carried by the bloodstream's constant motion. The brain's relationship with glucose is particularly tender. Despite making up only a small portion of the body's total mass, the brain consumes a large share of its energy, relying almost exclusively on glucose during normal conditions. This reliance means that the body prioritizes the brain's access to glucose above all else. Even when glucose levels drop in the blood, the body redirects what remains toward the brain, preserving cognitive function and awareness. This prioritization is part of the body's deeper instinct to maintain consciousness and internal regulation. The gentle movement of glucose into neurons supports the electrical activity that underlies sensation, thought, and calm awareness, illustrating how essential this molecule is to the experience of being alive. For tissues not currently active, glucose serves as a quiet potential. Glycogen stored in muscles and the liver becomes a form of reassurance, a reminder that energy is available even when external food sources are not. This storage capacity reflects the body's deep wisdom, a way of preparing for times when nourishment may be sparse. Because glycogen can be broken down quickly, it provides a buffer that keeps energy flowing smoothly across hours of rest or periods of fasting. This capacity for storing glucose gently underscores the body's desire for stability, safeguarding the steady hum of metabolism. As the bloodstream continues to carry glucose to its many destinations, the body remains attentive to small fluctuations. If glucose begins to rise again, insulin steps in. If it begins to fall, glucagon or other hormones respond. This interplay creates a calm internal dance, one that has been refined over millions of years of evolution. It ensures that glucose, though small and simple as a molecule, becomes part of a vast and gentle cycle that includes digestion, absorption, transport, storage, and use. In this balanced flow, glucose becomes more than just a nutrient. It becomes a symbol of the body's quiet intelligence. The movement of glucose through the bloodstream is not dramatic or loud. Instead, it reflects a deep harmony between organs, hormones, and cells, all working together to sustain energy in life. As the journey continues, the next part will explore what happens once glucose enters the cell and begins the first steps of energy production through glycolysis. But for now, it is enough to appreciate the calm elegance of glucose absorption and transport. 7. Glycolysis. The first quiet steps of energy release. When glucose finally crosses the threshold into a cell, its journey enters a new and intimate phase, one that unfolds not in the broad pathways of digestion or the gentle currents of the bloodstream, but in the quiet interior of the cell itself. Here, in the soft glow of the cytosol, glucose meets the series of reactions that will begin to unlock the energy it carries. This pathway is called glycolysis, and though its name may sound technical or distant, the process it describes is ancient, universal, and tenderly essential. Glycolysis is the first whisper of energy release, the first steady turning of molecular gears that support every heartbeat, every breath, and every moment of thought or rest. It is a process that occurs in every living cell, from the most delicate neuron to the simplest bacterium, unfolding with unbroken continuity as long as life persists. Glycolysis begins simply, with a single molecule of glucose resting inside the cell. Although glucose contains a reservoir of energy, this potential is locked within its bonds, inaccessible until those bonds can be rearranged. Rather than breaking them all at once, the cell approaches this task carefully. The first steps of glycolysis require a small investment of energy, a gentle push that encourages glucose to begin its transformation. This investment comes in the form of ATP, the cell's universal energy currency. At first, this may seem counterintuitive, using energy to generate energy, but the logic becomes clear when viewed through the calm sensibility of cellular processes. By spending this small amount of ATP, the cell primes glucose, making it easier to split and reorganize later in the pathway. The cell adds a phosphate group to glucose, turning it into glucose 6-phosphate. This small modification serves more than one purpose. It prevents glucose from drifting back out of the cell, anchoring it safely inside. It also begins the process of shaping the molecule for the reactions to come. In the next steps, another phosphate is added, creating a molecule known as fructose 1,6-bisphosphate. These initial transformations require patience and precision, as enzymes guide the molecules along, each interaction unfolding smoothly, almost like a slow turning of pages in a story that has been told countless times across the history of life. Once glucose has been prepared in this way, the pathway takes a subtle but important shift. The 6-carbon molecule is gently split into 2 smaller 3-carbon molecules. These are called glyceraldehyde 3-phosphate, and their formation marks the beginning of energy release. Though these molecules are identical, each one undergoes its own sequence of reactions, doubling the cell's eventual energy gain. The calm symmetry of this moment reflects the efficiency and quiet ingenuity of nature, where even a single molecule can be divided into mirrored forms that contribute to life with equal grace. As glycolysis continues, the cell begins to harvest energy, not in sudden bursts, but in small, measured amounts. Enzymes guide each reaction, removing electrons and transferring them to carrier molecules that will later deliver their energy to other pathways. These carrier molecules, such as NADMIs, act like gentle couriers, accepting electrons with ease and becoming NADH. This transformation represents a promise of future energy, stored temporarily until it can be released in later stages of metabolism. At the same time, ATP begins to appear again. Unlike the early steps, where ATP was spent, the later phases of glycolysis produce more than was invested. The cell gains a net total of 2 ATP molecules for each glucose molecule that completes glycolysis. This may not seem like much, but it is enough to support cells that can only rely on glycolysis for energy. Red blood cells, for example, lack mitochondria and therefore depend entirely on glycolysis for their ATP. Even neurons and muscle cells rely on glycolysis during moments of immediate need, when oxygen availability or sudden demand makes rapid energy necessary. Glycolysis answers this need with gentle consistency. Another product of glycolysis is pyruvate, the 3-carbon molecule that represents the final step of this pathway. Pyruvate sits at a crossroads, its fate determined by the cell's oxygen supply and metabolic demands. When oxygen is abundant, pyruvate travels to the mitochondria, entering the citric acid cycle and leading to the creation of much larger amounts of ATP. When oxygen is limited, such as during intense bursts of activity, pyruvate may be converted into lactate, allowing glycolysis to continue even under anaerobic conditions. This flexibility reflects the adaptability of cellular metabolism, a soft and responsive shifting of pathways that ensures energy is always available. The setting in which glycolysis takes place adds to its quiet nature. Unlike later stages of metabolism that occur within the mitochondria, glycolysis unfolds in the cytosol, the open, fluid space inside the cell. This environment is gentle and spacious, allowing enzymes and molecules to drift and interact without confinement. The reactions of glycolysis happen one after another, not through force, but through the natural affinity of molecules for one another. Enzymes bring reactants together, lowering the energy needed for each step, guiding transformations with a light touch that reflects millions of years of refinement. Perhaps one of the most calming aspects of glycolysis is its universality. Every organism on Earth, from the smallest microbes to the largest mammals, relies on glycolysis in some form. It is one of the earliest metabolic pathways to have evolved, a reflection of life's origins in simple environments that lacked oxygen. This ancient quality gives glycolysis a timelessness, a sense of continuity that stretches across evolutionary history. When glucose is slowly transformed inside your cells, it follows the same path once taken by the earliest living beings on the planet. This connection offers a peaceful perspective. The energy flowing through your cells has followed a pattern older than mountains, rivers, or stars as we know them. Despite its age, glycolysis remains essential because of its adaptability. When cells require quick energy, glycolysis is always ready, operating with steady speed regardless of oxygen levels. When energy demands shift or fluctuate, glycolysis adjusts quietly. During sleep, it keeps the body's resting tissues supplied with ATP. During physical activity, during fasting, it collaborates with other metabolic processes, ensuring the brain and other organs still receive enough energy to function calmly. What emerges from glycolysis is not simply ATP or pyruvate. It is a soft foundation upon which all other energy pathways depend. Without glycolysis, cells would have no entry point into metabolism, no initial process to begin the release of energy. It is the slow unlocking of a door that leads to deeper chambers of biochemical activity. Its simplicity is its strength, and its quietness is its elegance. As pyruvate forms and the final steps of glycolysis come to a close, the pathway gently hands its products forward to whatever processes the cell requires next. It is not concerned with what follows. Its task is complete. And yet, without this beginning, the more complex stages of metabolism, the citric acid cycle, and oxidative phosphorylation, could not occur. Glycolysis is the calm first act in the body's energy story, a story that continues in the mitochondria with deeper and more elaborate steps. As we move into the next part of this journey, the focus will shift toward these mitochondrial pathways, where pyruvate enters a new realm and energy production grows more expansive. But before taking that step, it is worth resting for a moment in the simplicity of glycolysis, the elegant, gentle series of reactions that sustains life in its earliest and most immediate form. 8. Pyruvate processing and entry into the mitochondria. After glycolysis completes its quiet sequence of gentle reactions, the cell is left with pyruvate, a small and unassuming 3-carbon molecule that carries within it the next chapter of energy transformation. Pyruvate rests at a crossroads where the cell evaluates its surroundings with the soft precision that characterizes all of metabolism. The decision the cell makes in this moment is not abrupt or forceful. It is instead shaped by the presence or absence of oxygen, by the subtle shifts in energy demand, and by the gentle hormonal signals that guide the body's internal rhythms. When oxygen is available as it is during calm breathing, steady movement, or restful states, the pyruvate pathway moves forward into a space where energy production grows deeper and more sustained. This space is the mitochondrion, often described as the cell's powerhouse, though its actions are far more serene and intricate than that simple phrase suggests. Before pyruvate can enter the mitochondria, it must undergo a small but essential transformation. This modification occurs in the mitochondrial matrix, but the process begins as pyruvate approaches the outer membrane. The mitochondrion has 2 membranes, outer and inner, each with its own character. The outer membrane is permeable to small molecules, allowing pyruvate to pass through easily. The inner membrane, however, is more selective. It allows entry only through specific transport proteins that usher molecules inward with calm intention. Pyruvate travels through one such carrier, slipping into the mitochondrial matrix where a welcoming environment awaits. Pyruvate dehydrogenase complex resides, a group of enzymes working together in a coordinated fashion that resembles a slow, graceful dance. The pyruvate dehydrogenase complex is responsible for converting pyruvate into acetyl-CoA, the molecule that will enter the citric acid cycle. This conversion is not merely a chemical reaction, it is a gateway, a gentle unveiling of deeper energy reserves. In this step, one carbon is removed from pyruvate in the form of carbon dioxide, a small and harmless gas that easily diffuses out of the mitochondrion and is eventually carried to the lungs for release. What remains of pyruvate, now a 2-carbon fragment, binds with coenzyme A, forming acetyl-CoA. This molecule represents the start of a new phase of metabolism, one that will unfold within the citric acid cycle and contribute to the gradual release of high-energy electrons. The conversion of pyruvate to acetyl-CoA also produces NADH, a gentle carrier of electrons that will later participate in oxidative phosphorylation. NADH holds onto these electrons delicately, keeping their energy safe until it is time to release it through the electron transport chain. In this way, pyruvate processing is not just a transition step, it is the preparation of essential components that support the cell's deeper energy needs. The cell does not hurry this process. It takes place smoothly, with each enzyme contributing its careful movement to the transformation. Within the mitochondria, the environment is warm, carefully regulated, and filled with enzymes arranged in a way that feels almost architectural. The shape of the inner membrane with its folds known as cristae maximizes the surface area for reactions that will occur later. These folds create pockets and pathways, giving mitochondria the unique ability to manage energy production efficiently. When pyruvate enters this space, it becomes part of a larger system, a network of reactions that reflects the quiet intelligence of cellular design. As acetyl-CoA forms, it gently joins the citric acid cycle, also called the Krebs cycle or the tricarboxylic acid cycle. This cycle is not a rapid burst of chemical activity, but a continuous, looping series of reactions where energy is gathered in small, steady increments. Acetyl-CoA binds with oxaloacetate, forming citrate, and from this point the cycle unfolds, producing more NADH, FADH2, and a small amount of ATP. But the importance of acetyl-CoA extends beyond the citric acid cycle. This molecule also participates in lipid metabolism and serves as a precursor for building other essential compounds. In this sense, pyruvate processing is not only an energy-producing step, but also a moment where the cell decides how best to use its resources, whether for fuel, structure, or storage. The reason pyruvate must undergo this careful transformation becomes clearer when considering the nature of metabolism itself. Glucose, as helpful as it is, cannot directly enter the citric acid cycle. It is too large, too energy-dense in its original form. Pyruvate bridges the gap between glycolysis and the more advanced stages of energy extraction. By converting pyruvate into acetyl-CoA, the cell creates a molecule that fits precisely into the cycle's architecture, allowing energy to be released evenly and safely. The removal of one carbon atom ensures that the molecule remains balanced for the reactions to come. Each step is measured, thoughtful, and aligned with the body's broader need for stability. The presence of oxygen influences this process in a profound way. When oxygen is plentiful, the mitochondria can proceed through the full sequence of energy production, including oxidative phosphorylation, which produces the most ATP. Oxygen serves as the final electron acceptor in the electron transport chain, ensuring that NADH and FADH2 can release their electrons and return to their oxidized forms. This allows glycolysis, pyruvate processing, and the citric acid cycle to continue without interruption. In the absence of oxygen, however, the pathway shifts. Pyruvate cannot enter the mitochondria under anaerobic conditions because the electron carriers would remain in their reduced forms, creating a bottleneck in the system. Instead, pyruvate is converted into lactate in the cytosol, allowing glycolysis to keep producing a small amount of ATP. This alternative pathway is temporary and reversible, designed to support the body during moments of sudden exertion or limited oxygen availability. When oxygen returns, lactate can be converted back into pyruvate or used by the liver to regenerate glucose. Despite these shifts, the central pathway remains the same. When oxygen is available, pyruvate enters the mitochondria, undergoes its gentle transformation, and becomes acetyl-CoA. This molecule represents a convergence point where carbohydrates, fats, and proteins meet. Fatty acids can be converted into acetyl-CoA through beta oxidation, while certain amino acids enter the cycle at this same step. This convergence reflects the unity of metabolism, showing that different types of nutrients ultimately contribute to the same energetic crossroads. Whether the cell draws from glucose, fats, or proteins, the final pathways share a common structure, emphasizing the elegant interconnectedness of life's chemistry. The entry of pyruvate into the mitochondria is not only about energy, it also symbolizes the cell's quiet decision-making, its ability to assess conditions, adjust pathways, and ensure that energy flows in a way that supports both activity and rest. In this way, metabolism mirrors the broader rhythms of living, moments of higher demand followed by moments of calm balance. Pyruvate processing helps maintain this internal harmony, ensuring that energy is neither wasted nor withheld, but released gently as needed. As the final molecules of acetyl-CoA are formed and the mitochondrial pathways prepare to continue their work, the story of energy transformation deepens. The next chapter will explore the citric acid cycle itself, the slow, circular pathway that lies at the center of metabolism. But before moving ahead, it is worth pausing to appreciate the subtlety of this step. Pyruvate processing is the quiet threshold between the simple beginnings of glycolysis and the intricate world within the mitochondria, a threshold crossed seamlessly, molecule by molecule, in the soft hum of living cells. 9. The citric acid cycle, a circular flow of intermediates. When acetyl-CoA enters the citric acid cycle, it steps into one of the most elegant and enduring patterns of biochemical life. This cycle, nestled deep within the mitochondrial matrix, it turns with the calm assurance of a process that has been refined over billions of years. A circular flow that repeats gently with every breath you take, every moment you rest, and every quiet beat of the heart. The citric acid cycle is not a dramatic event. It is a soft, rhythmic sequence of reactions in which molecules meet, part, and transform, all to release the stored energy that nourishes the body. Through this cycle, energy becomes available. Carbon dioxide is released with each exhale, and electron carriers are filled, ready to deliver their precious cargo to the next stage of energy production. The cycle begins when acetyl-CoA merges with oxaloacetate, a 4-carbon molecule that patiently waits for each new arrival. Together they form citrate, a 6-carbon molecule whose shape and structure mark the beginning of a fresh turn around the pathway. This first joining is simple but profound, for citrate is the foundation of the cycle's unfolding. The creation of citrate represents the merging of the food you eat with the inner machinery of your cells, a moment in which nourishment becomes part of your own biological rhythm. Oxaloacetate, though temporarily transformed, will eventually be regenerated at the end of the cycle, ready to greet another acetyl-CoA molecule in a continuous flow that never stops as long as the cell has energy needs. Once citrate is formed, it undergoes a gentle rearrangement into isocitrate. This shift is not forceful. It is merely a repositioning, a subtle turning of the molecule that prepares it for the steps that follow. Enzymes guide these changes with careful precision, making sure each reaction unfolds smoothly. The enzymes themselves are quiet catalysts, always present, never consumed, always guiding, never commanding. They encourage molecules to transition into new shapes and forms, creating the conditions for energy to be released in soft increments rather than in sudden bursts. As isocitrate moves forward, it begins to release carbon dioxide. This step, like the exhalation that accompanies it, is natural and unhurried. One carbon molecule departs, carried away gently through the bloodstream to the lungs, where it will eventually leave the body with each gentle breath. During this transformation, electrons are also released, captured by NAD mice, which becomes NADH. These NADH molecules are like subtle energy messengers, storing the high-energy electrons that will later be used to power the electron transport chain. Each NADH produced during the citric acid cycle is a quiet promise of energy yet to be fully realized. The next phases of the cycle continue this pattern. Each transformation removes another carbon. Each electron transfer fills another carrier, and each small shift brings the cycle closer to regeneration. The molecule at the center of these transitions gradually becomes smaller again, losing the carbon that originally came from acetyl-CoA. With every release of carbon dioxide, the cycle becomes a reflection of respiration itself, a gentle pairing of inhalation and exhalation, of oxygen entering the body and carbon leaving. In this sense, the citric acid cycle mirrors the act of breathing, not in its physical form, its essence, a continuous exchange that sustains life with each turn. As the molecule continues along its path, it becomes succinate, then fumarate, then malate. Each of these intermediates is unique, yet each one flows naturally into the next, like stepping stones placed carefully across a slow-moving stream. The transitions are subtle. Electrons shift, bonds reorganize, water molecules come and go, adding stability or guiding shape as needed. FADH, too, another electron carrier, is produced along the way, holding on to its electrons as gently as NADH, ready to contribute them to the next phase of energy release. The cycle concludes with the regeneration of oxaloacetate, the same molecule that began the sequence. This return creates a sense of continuity, a reminder that in the world of biochemistry, cycles are not simply processes, they are rhythms of existence, repeating patterns that reflect the balanced nature of life. The regeneration of oxaloacetate ensures that the cycle can continue endlessly, provided that the cell has acetyl-CoA to feed into it. This continuous movement means that at any given moment, countless mitochondria within your body are turning through this cycle, step by step, molecule by molecule, contributing to the gentle hum of energy that sustains you. The purpose of the citric acid cycle is not to produce large amounts of ATP directly. Instead, its purpose is to fill electron carriers like NADH and FADH2. These carriers hold the energy extracted from acetyl-CoA and transport it to the electron transport chain. In this way, the citric acid cycle is a preparatory pathway, carefully gathering energy in forms that can later be released with greater magnitude. Without this cycle, the cell would not have the steady supply of electron carriers needed for effective oxidative phosphorylation. The cycle itself releases only a small amount of ATP, but it is the generous amount of NADH and FADH2 it generates that fuels the next stage. The circular nature of the cycle reflects an important truth. Life moves in patterns of renewal. Energy is never taken all at once, but is released slowly, in stages, through pathways that preserve stability. The citric acid cycle embodies this philosophy beautifully. It gathers, releases, transforms, and regenerates, maintaining a delicate equilibrium that serves the cell and sustains the body. The calm repetition of this cycle ensures that no matter your state awake or asleep, active or resting, the mitochondrial energy pathways continue their quiet support. Another soft beauty of the citric acid cycle is its interconnectedness with other metabolic pathways. Carbohydrates feed into it through acetyl-CoA. Fatty acids, broken down through beta oxidation, enter the cycle in the same way. Certain amino acids can also supply intermediates, joining the cycle at various points. Because of this, the citric acid cycle acts as a meeting ground for all three major nutrients, carbohydrates, fats, and proteins blending their contributions into a single harmonious flow. This unity demonstrates that metabolism is not a set of isolated pathways, but a deeply interwoven system where everything supports everything else. The citric acid cycle also produces intermediates that can be used for biosynthesis. Cells may draw from the cycle to create amino acids, fatty acids, or other molecules as needed. When this happens, the cell quietly replenishes those intermediates from other sources, maintaining the cycle's continuity. This gentle balancing act is an example of the body's persistent wisdom, always adjusting, always compensating, always tending to the needs of each cell. As this cycle continues, turning softly, releasing energy in manageable pieces, and preparing molecules for the deeper stages of metabolism, it remains a symbol of biochemical calm, a predictable, rhythmic motion that supports life without ever disturbing it. With each turn, electron carriers are filled, carbon dioxide is released, and energy becomes more accessible. In the next part, the journey will move into oxidative phosphorylation, where the electrons carried by NADH and FADH2 are used to create a larger flow of ATP. This final stage of carbohydrate energy release is both powerful and gentle, driven by gradients and quiet molecular currents. But before stepping into that realm, it is soothing to sit for a moment with the calm continuity of the citric acid cycle, the soft circular pathway at the heart of metabolism. 10. Oxidative phosphorylation, a calm cascade of energy formation. As the citric acid cycle completes its gentle turn and hands off its filled electron carriers, the story of energy transformation moves into its most refined and delicate chapter: oxidative phosphorylation. This process, unfolding in the inner membrane of the mitochondria, is where the bulk of the cell's ATP is formed. Yet despite its importance, oxidative phosphorylation is not a violent or forceful release of energy. It is a quiet cascade, a slow, steady flow of electrons and protons that creates the soft currents needed to power life. It moves with the precision of a tide and the serenity of a deep breath, reminding us that even the most essential cellular processes unfold in peaceful, rhythmic steps. The inner mitochondrial membrane, with its many folds called cristae, provides the perfect landscape for oxidative phosphorylation. These folds increase the surface area available for the protein complexes that guide electrons along their journey. Embedded within this membrane are 4 large complexes, along with a few smaller carriers, all aligned to receive electrons from NADH and FADH2. These carriers are like travelers coming from the citric acid cycle, holding onto the electrons they collected earlier. When they arrive at the inner membrane, they do not release these electrons abruptly. Instead, the electrons enter the electron transport chain, flowing gently from one complex to the next. The electron transport chain operates with careful choreography. NADH donates its electrons to the first complex, while FADH2 donates its electrons slightly later in the chain. These electrons move through the complexes step by step, guided by the differences in energy levels between them. At each step, a small amount of energy is released, not enough to disrupt the cell, but enough to support the next phase of the process. The movement of electrons is quiet and continuous, like a small stream flowing over stones, carrying with it the potential to create something far greater downstream. As electrons pass through the complexes, the energy they release is used to pump protons, hydrogen ions, across the inner mitochondrial membrane, from the matrix into the intermembrane space. This creates a gradient, a difference in concentration between the two sides of the membrane. The matrix becomes relatively calm and low in protons, while the intermembrane space becomes enriched with them. This gradient is not tense or unstable. Instead, it functions like a gentle reservoir of potential energy, a quiet pressure that builds slowly and equally until the moment it is released. This proton gradient is often described as a proton motive force, but any sense of force here is soft and measured. The gradient does not burst through the membrane. It does not drive reactions abruptly. Instead, protons flow back into the matrix through a remarkable protein called ATP synthase. ATP synthase rests within the membrane like a small turbine, awaiting the flow of protons to turn its components. As protons move through, the protein rotates with a calm, rhythmic motion, performing a subtle mechanical action that allows it to bind ADP and inorganic phosphate, joining them together to form ATP. The creation of ATP is gentle and precise. Every turn of ATP synthase brings new molecules together, shaping them into the fundamental currency of energy that cells use for nearly every task. The ATP formed here supports muscle relaxation, brain activity, cellular repair, protein synthesis, and the maintenance of ion gradients. It fuels the quiet movement of cilia, the steady beat of the heart, and the slow turnover of cellular materials. Though ATP is created in large quantities, the process that forms it remains completely smooth and continuous. As the electrons reach the end of the electron transport chain, they meet oxygen, the final electron acceptor. This moment is subtle but essential. Oxygen receives the electrons and combines with protons within the matrix to form water. This reaction completes the chain and ensures that electrons can continue flowing smoothly. Oxygen's role here feels almost symbolic, as if representing the final, peaceful closing of a cycle. Without oxygen to accept the electrons, the chain would stall, proton pumping would cease, and ATP production would slow. But as long as oxygen is available, the cascade continues, unbroken. Water, produced through this process, is not wasteful or excessive. It becomes part of the cell's environment, contributing to hydration and supporting additional reactions throughout the mitochondria. This is one of the reasons why breathing and energy production are so deeply intertwined. Every breath gently delivers oxygen to the bloodstream, which brings it to the cells, and the cells in turn use it to complete this quiet and essential pathway. In this sense, oxidative phosphorylation is synchronized with the act of breathing, creating a natural rhythm between the external world and the internal world of the cell. The proton gradient that drives ATP synthase is one of the most elegant aspects of biology. Instead of releasing energy all at once, the cell uses the gradient to store potential energy safely and release it gradually. This slow release supports life with stability. The body is not designed for sudden shifts in energy. It requires consistency, balance, and smooth transitions. Oxidative phosphorylation provides exactly this. The proton gradient rises and falls gently with the pace of metabolism, responding to changes in cellular demand with subtle adjustments. During moments of rest, the pathway still operates, but at a slower rhythm. Energy needs decrease, and ATP synthesis continues just enough to sustain essential functions. During activity, the pathway increases its pace, pumping more protons and creating more ATP, but even then it does so without harsh transitions. The mitochondria sense the need and quietly adjust their actions to match. It offers the cell a steady supply of energy. Whether in times of calm or exertion, the interconnectedness of oxidative phosphorylation with other energy pathways deepens the sense of harmony. The NADH and FADH2 produced in glycolysis and the citric acid cycle deliver their electrons to this process. Their work is not complete until oxidative phosphorylation has converted their stored energy into usable ATP. In turn, oxidative phosphorylation depends on the earlier pathways to supply these carriers. No single part of metabolism exists in isolation. All are woven together in a quiet tapestry of continuous activity. As ATP is produced and released into the matrix, it is transported out of the mitochondria into the cytosol, where it becomes available to power cellular functions. Another set of transporters ensures that newly formed ATP leaves the mitochondria smoothly while ADP re-enters, ready to be recycled by ATP synthase. This transport occurs with a kind of gentle efficiency, ensuring that ATP never accumulates excessively and that ADP is always available to continue the cycle. Even in this, the cell maintains balance, preventing any interruptions in the flow of energy. Oxidative phosphorylation represents the climax of energy production, yet it remains completely calm in its action. There is no rapid combustion, no abrupt release, no sudden surge of energy. Instead, electrons move quietly, protons drift softly, ATP synthase turns steadily, and oxygen sits at the end like a peaceful receptor, accepting electrons to keep the chain moving forward. This pathway is the quiet engine of life, allowing cells to work with grace and subtlety. As we continue the journey through metabolism, the next chapter will move from carbohydrates to lipids, those long-lasting reservoirs of energy stored throughout the body. But before stepping into that new landscape, it is soothing to appreciate the serene mechanics of oxidative phosphorylation, the gentle cascade that turns the energy of electrons into the sustaining warmth of ATP. 11. Lipids, concentrated stores of slow, sustained energy. Among all the nutrients the body receives, lipids hold a special and quietly powerful place. They are the deep reservoirs of energy, softly stored away in tissues where they rest patiently until the body calls on them. Unlike carbohydrates, which offer quick bursts of energy that rise and fall gently through the day, lipids provide something slower, steadier, and more enduring. Their nature reflects calm endurance energy woven into long chains, stored with care, waiting for the moments when the body needs it most. Whether carried in the oils of plants, the fats of animals, or the membranes of cells themselves, lipids offer a richness of structure and function that supports life in subtle and essential ways. Lipids come in many forms, each with its own unique character. Triglycerides, the most familiar form, consist of 3 fatty acids bound to a glycerol backbone. They rest within adipose tissue, forming droplets that the body can access when energy runs low. Phospholipids, arranged in layers, create the quiet architecture of cell membranes, thin, flexible boundaries that hold each cell together. Cholesterol, often misunderstood, lends stability to these membranes and serves as a precursor to hormones and vitamins. Even among fatty acids, there is variety. Some are saturated, forming straight chains that pack closely together. Others are unsaturated, bending gently where double bonds introduce a soft flexibility. This diversity allows lipids to serve roles that range from structural to metabolic, offering support to every system in the body. When lipids enter the digestive system, the body approaches them with a different pace than it uses for carbohydrates. Fats are not water-soluble, and because of this, they move through the digestive tract with calm persistence. In the stomach, fats separate naturally from the rest of the chyme, forming small droplets that float gently within the warm mixture. The stomach does not break them down entirely, but it does prepare them, churning slowly and allowing gastric lipase to begin the early stages of digestion. This enzyme works quietly, releasing a few fatty acids and softening the triglycerides so that the next steps can unfold smoothly. As this mixture enters the small intestine, a remarkable transformation takes place. The liver releases bile, an emerald green fluid stored in the gallbladder and delivered to the intestine precisely when needed. Bile does not break fats down chemically, but instead emulsifies them. It coats the fat droplets with bile salts, causing them to disperse into smaller and smaller droplets. This process greatly increases the surface area available to digestive enzymes. It is a gentle unfolding, a turning of large shapes into tiny ones that drift freely in the watery environment of the intestine. With emulsification complete, pancreatic lipase can move in with ease, cleaving triglycerides into monoglycerides and free fatty acids. These smaller molecules gather together into structures called micelles, tiny spherical arrangements created by the mutual cooperation of bile salts and lipids. Micelles drift slowly toward the brush border of the intestinal lining, bringing their contents close enough for absorption. Fatty acids and monoglycerides leave the micelles effortlessly and slip into the enterocytes, the cells that line the small intestine. Once inside, another transformation takes place. The fatty acids and monoglycerides are reassembled into triglycerides, demonstrating the body's quiet efficiency in handling nutrients. These newly rebuilt triglycerides join with proteins, cholesterol, and phospholipids to form chylomicrons, large and delicate particles designed to transport fats through the aqueous environment of the body. Chylomicrons do not enter the bloodstream immediately. Their size leads them instead into the lymphatic vessels, where they travel at an unhurried pace through the lymphatic system. This system moves slowly, driven not by the pulse of the heart but by the gentle contraction of surrounding muscles. Eventually, the chylomicrons reach the bloodstream through the thoracic duct, delivering lipids throughout the body with a softness that mirrors the calm nature of the nutrients they carry. Tissues such as adipose, muscle, and the liver receive these lipids, storing or using them as needed. Adipose tissue, spread in quiet pockets throughout the body, is the primary reservoir for lipid storage. Within these cells, triglycerides form droplets that expand or contract depending on the body's energy needs. These droplets are not rigid or static. They are dynamic, responding to hormonal cues that tell them when to save energy and when to release it. Insulin encourages adipose cells to store triglycerides after a meal, while hormones such as glucagon and epinephrine gently signal the release of fatty acids during fasting or physical activity. This balance reflects a peaceful harmony between nourishment and expenditure, allowing the body to maintain a soft continuity of energy over hours, days, and even longer periods. When the body needs to use these stored lipids, a process called lipolysis begins. Enzymes within adipose cells break triglycerides into free fatty acids and glycerol. These molecules slip into the bloodstream, carried by albumin, a protein that escorts them to tissues requiring energy. Muscle cells, the liver, and many other tissues take in these fatty acids with ease. Once inside, the fatty acids are transported to mitochondria, where they undergo beta oxidation, a stepwise breakdown that produces acetyl-CoA, NADH, and FADH2. These products feed directly into the citric acid cycle and oxidative phosphorylation, allowing lipids to contribute to ATP formation in a way that is both efficient and deeply sustaining. The energy provided by lipids is notably slow and steady. Fatty acids produce far more ATP per molecule than carbohydrates because their long chains hold more potential energy. This makes lipids ideal for periods when energy demands are extended, such as during prolonged fasting, endurance activities, or quiet overnight. Metabolism. While glucose offers quick support, lipids provide the enduring background hum of energy that keeps the body functioning, even without frequent meals. Their presence reflects the body's preference for balanced short-term and long-term fuels working together to maintain harmony. Beyond their role as energy sources, lipids support life in countless subtle ways. They form the cell membranes that protect and define every cell. These membranes are not rigid walls. They are flexible, shifting boundaries that allow cells to communicate, respond, and adapt. Phospholipids create a bilayer that holds cellular contents while allowing essential molecules to pass through. Cholesterol weaves itself among these phospholipids, providing stability and adjusting the membrane's fluidity in response to temperature changes. Without lipids, the structure of life itself would be impossible. Every heartbeat, every nerve signal, every hormonal message depends on the delicate architecture provided by these molecules. Lipids also serve as the foundation for signaling molecules, such as prostaglandins, which help regulate inflammation, blood flow, and many other processes. Steroid hormones, including cortisol, estrogen, and testosterone, are synthesized from cholesterol, illustrating yet another role lipids play in maintaining balance across the body. Even the brain, with its intricate networks and quiet electrical activity, relies heavily on lipids. Much of its structure is made from fatty layers that insulate nerve fibers, ensuring that signals travel softly and efficiently. In this way, lipids support not only bodily energy, but also perception, thought, and emotion. Their presence in the diet is equally gentle. Lipids provide texture, flavor, and satisfaction to foods. They slow the movement of food through the digestive tract, allowing for more gradual absorption and creating a feeling of fullness that aligns with the body's natural pacing. They carry fat-soluble vitamins A, D, E, and K, making it possible for these essential nutrients to be absorbed and used. Each contribution reflects a sense of completeness, showing that lipids are not simply stored energy but companions to many aspects of nourishment. As lipids weave through these roles, they remind us that energy in the body is not a single event or a single pathway. It is a continuum, a soft layering of processes that support each other. Lipids offer one of the slowest and most enduring layers, providing deep reserves of energy and structural stability. Their transformations occur quietly, whether through digestion, transport, storage, or oxidation, and as they move through the body, they enhance the sense of continuity and grounding that underlies all biological processes. In the next part of this journey, we will follow fatty acids more closely as they travel deeper into energy pathways, exploring the calm precision of beta oxidation and the ways lipids become integrated into cellular metabolism. But for now, it is enough to rest with the understanding that lipids serve as the body's long, steady threads of energy, always present, always patient, always supporting the quiet flow of life. 12. Fat digestion and transport through the body. When fats enter the body, they begin a journey that is slower and more delicate than that of carbohydrates or proteins. Fat digestion unfolds in soft, unhurried movements, shaped by the unique nature of lipids themselves. Because fats do not dissolve easily in water, they glide through the digestive tract in gentle droplets, untouched by the watery environment that surrounds them. Their processing requires a series of quiet, carefully coordinated steps: emulsification, enzymatic cleavage, absorption, and transport through the lymphatic system. Each step is designed not to rush the fats along, but to guide them patiently toward a form the body can use, store, or transform. This journey begins in the mouth, though only lightly. Lingual lipase, an enzyme released in the saliva, starts breaking down triglycerides even before the food is swallowed, but its activity is subtle. In the stomach, gastric lipase takes over with calm persistence. Yet most fat digestion occurs beyond the stomach. Here, in the darkness and quiet warmth of the gastric cavity, fats simply prepare for what is to come, blending with the rest of the meal and forming a smooth mixture that will eventually make its way into the small intestine. As this mixture passes through the pyloric sphincter, it enters the duodenum, the beginning of the small intestine, where a remarkable transformation takes place. The presence of fats signals the gallbladder to release bile, a greenish gold fluid made in the liver. Bile does not digest fats chemically, but instead performs a different kind of work. It coats the fat droplets, breaking them into smaller and smaller pieces in a process called emulsification. This step is essential because enzymes can digest fats efficiently only when the surface area is increased. The bile salts arrange themselves around each droplet, forming a delicate barrier that keeps the droplets from merging back together. The result is a fine suspension of tiny fat globules, each one drifting independently in the watery environment of the intestine. With the fat now emulsified, pancreatic lipase begins its gentle, skillful work. Released from the pancreas in response to hormonal signals, lipase attaches itself to the droplets and cleaves triglycerides into free fatty acids and monoglycerides. These smaller molecules remain surrounded by bile salts, forming micelles, tiny, spherical clusters that serve as carriers. Micelles move slowly toward the brush border of the intestinal lining, ferrying their contents like small boats drifting toward shore. Their movement is not forced. It is guided by natural gradients and the rhythmic contractions of the intestine, patterns of motion that reflect the body's quiet pacing. When the micelles reach the intestinal cells, they release their contents. Fatty acids and monoglycerides slip through the cell membrane and enter the enterocytes, the specialized cells that line the small intestine. Once inside these cells, the transformation continues. Instead of remaining as separate molecules, these components are reassembled back into triglycerides. This rebuilding might seem strange at first. After all, the body just spent effort breaking triglycerides down, but this sequence serves a purpose. The breakdown allows fats to enter the cells, and the rebuilding ensures they can be packaged for transport in a way the body finds safe and efficient. To prepare these triglycerides for travel, the enterocytes wrap them in a coating of proteins, cholesterol, and phospholipids, forming large, spherical particles known as chylomicrons. These particles are much too large to enter the bloodstream directly, and for that reason, they take a different route. Instead of slipping into capillaries, they enter the lacteals, the small lymphatic vessels that run through each intestinal villus. The lymphatic system moves slowly, driven by gentle muscular contractions and the natural rhythm of the body. Instead, it carries its contents forward in a serene, almost drifting motion, perfectly suited to the delicate structure of chylomicrons. As the chylomicrons travel through the lymphatic system, they eventually reach the thoracic duct, a large channel that empties into the bloodstream near the heart. Here, the chylomicrons enter circulation for the first time. This slow entry ensures that fats do not overwhelm the bloodstream, but instead become available gradually, providing a steady supply of nutrients to tissues that need them. Once in the bloodstream, chylomicrons begin to circulate throughout the body. Tissues such as muscle and adipose tissue express an enzyme called lipoprotein lipase on their capillary walls. This enzyme recognizes passing chylomicrons and begins to gently remove fatty acids from them. These fatty acids enter the nearby cells, where they can be used immediately for energy or stored as triglycerides for future needs. In muscle cells, they can be oxidized to produce ATP, supporting movement, posture, and the subtle work of maintaining body temperature. In adipose tissue, they settle into storage droplets, resting quietly until hormonal signals call upon them during periods of fasting or increased energy demand. The chylomicrons themselves continue to shrink as they lose their fatty acids. What remains eventually returns to the liver, where the particles are dismantled and recycled. The liver sorts through their components, using the cholesterol for hormone synthesis, incorporating the phospholipids into membranes, or converting remnants into new types of lipoproteins that will carry fats internally produced by the body. No part of this process is hurried. It is a slow, thoughtful cycle of receiving, transforming, and redistributing nutrients. Meanwhile, the small intestine continues its careful role in digestion. Not all fats are absorbed in the same way. Short-chain and medium-chain fatty acids, found in foods such as dairy and coconut, often enter the bloodstream directly without the need for micelles or chylomicrons. Their smaller size allows them to pass straight into the portal vein, traveling quickly to the liver where they are metabolized with ease. This contrast illustrates how the body handles different types of fats with different levels of care, always choosing the path that maintains balance. The digestion and transport of fats reflect a broader theme in metabolism. Some forms of nourishment move swiftly, while others flow more slowly, providing stability rather than immediacy. Lipids belong to the latter group. They offer energy that can be drawn upon not just in the moment, but across hours, days, and even weeks. Their presence in chylomicrons, their travel through the lymphatic system, and their storage in adipose tissue all point toward a long-term view of energy. This slow pacing is mirrored in the body's hormonal control of fat digestion. The presence of fats in the small intestine releases hormones such as cholecystokinin, CCK, which signals the gallbladder to contract, the pancreas to release enzymes, and the stomach to slow its emptying. These signals ensure that digestion remains balanced, that nutrients enter the body gradually, and that the digestive system has the time it needs to process everything fully. Nothing about fat digestion is abrupt. It reflects patience, precision, and the body's innate understanding of timing. Transporting fats through the body is not just a matter of moving energy. It also serves structural and regulatory needs. Fat-soluble vitamins A, D, E, and K travel alongside dietary lipids, relying on micelles and chylomicrons to reach their destinations. These vitamins support vision, immunity, antioxidant protection, and blood clotting. Without the pathways created for lipid transport, these essential nutrients would remain out of reach. In this sense, the digestion and movement of fats represent not just an energy system but a broader network supporting many layers of well-being. As chylomicrons complete their journey, and as tissues receive the gentle nourishment they provide, the body maintains a continuous rhythm of digestion, absorption, and distribution. Each cycle reflects the quiet intelligence of metabolism, the way it adapts to the nature of each nutrient with methods that are both elegant and efficient. In the next part, we will follow fatty acids deeper into the cell, exploring the steady steps of beta oxidation and the way lipids slowly and calmly yield their stored energy. But for now, the quiet journey of fats through the digestive system offers a reminder of how the body balances nourishment with time, guiding each molecule along the pathways that allow life to unfold with softness and stability. 13. Beta oxidation. How fatty acids become steady energy. When fatty acids finally reach the interior of a cell, their long, quiet journey toward becoming energy enters a new and deeply orderly phase. Unlike glucose, which releases its energy swiftly through glycolysis, fatty acids move into a slower and more measured pathway. This process, known as beta oxidation, takes place inside the mitochondria, the same calm, folded structures that support the citric acid cycle and oxidative phosphorylation. Beta oxidation does not rush. It works in gentle, repeated steps, trimming fatty acids little by little, transforming them steadily into acetyl-CoA, NADH, and FATH2. Through these transformations, fatty acids become one of the most enduring sources of cellular energy, offering fuel that can last through long stretches of fasting, quiet nights of sleep, or extended periods of calm activity. Before beta oxidation can begin, fatty acids must first enter the mitochondria, a process that itself unfolds with patient precision. Short-chain and medium-chain fatty acids can pass through the mitochondrial membranes with ease, slipping inside without the need for any specialized transport. But long-chain fatty acids, those that carry the most stored energy, require a gentle guiding system called the carnitine shuttle. This shuttle does not push fatty acids aggressively. Instead, it picks them up and escorts them slowly through the inner membrane. Carnitine binds to the fatty acid, forming a temporary compound that can be recognized by a transport protein. Once safely across the membrane, the fatty acid is released. Carnitine returns to the outer membrane to collect another molecule, and the process repeats. This cycle continues quietly, ensuring a steady flow of fatty acids into the mitochondria. Once inside, the fatty acid undergoes activation, a small but essential commitment that prepares it for beta oxidation. This activation attaches coenzyme A to the fatty acid, creating fatty acyl-CoA. This step requires a bit of energy from ATP, a soft investment that allows the fatty acid to be processed efficiently. With activation complete, the fatty acyl-CoA enters the beta oxidation pathway, where each round of reactions removes a 2-carbon segment in the form of acetyl-CoA. This repeating pattern resembles a slow unraveling of the fatty acid chain, cutting it into small, usable pieces that feed into other metabolic pathways. Beta oxidation consists of 4 recurring reactions, each one catalyzed by enzymes that quietly perform their tasks without fanfare. The first step gently introduces a double bond into the fatty acyl-CoA molecule, producing FADH2 as electrons are transferred. This flavin-based electron carrier drifts toward the electron transport chain, where it will eventually support ATP production. The next step hydrates the molecule, adding water in a simple, graceful movement that prepares it for the next transformation. After hydration, the molecule is oxidized again, producing NADH, a second type of electron carrier filled with potential energy. Finally, the molecule is split, releasing an acetyl-CoA unit and leaving behind a slightly shorter fatty acyl-CoA. This shorter molecule re-enters the cycle, beginning the 4 steps again. This repeating rhythm continues until the entire fatty acid has been transformed into acetyl-CoA units. The length of the fatty acid determines how many rounds of beta oxidation occur. A 16-carbon fatty acid like palmitate undergoes 7 cycles and produces 8 acetyl-CoA molecules, providing far more energy than a similar number of carbons in a carbohydrate. This difference is one reason why lipids serve as such enduring reservoirs of energy. Their long chains hold energy densely, and beta oxidation releases it slowly and steadily, one small step at a time. As the acetyl-CoA molecules form, they slip into the citric acid cycle, merging seamlessly with the energy pathways created for carbohydrates. Here, they are oxidized further, producing more NADH and FADH, 2 that feed into oxidative phosphorylation. The electrons carried by these molecules contribute to the creation of proton gradients that drive ATP synthase, leading to the formation of large amounts of ATP. In this way, beta oxidation is not an isolated process but part of a larger, interconnected network that ensures energy is always available. The slow, steady supply of acetyl-CoA from fatty acids helps maintain a stable flow of energy. The calmness of beta oxidation reflects the nature of fats themselves. They are not meant to be used in quick bursts, but rather to serve as long-lasting reserves. When the body rests overnight, when meals come at long intervals, or when physical activity continues gently for extended periods, fatty acids become the preferred fuel. Their oxidation produces a steady stream of ATP that supports the body without causing sudden shifts in blood sugar or energy availability. Even the brain, though it relies primarily on glucose under normal circumstances, can indirectly benefit from this slow, steady supply during fasting, as the liver converts fatty acid byproducts into ketone bodies that the brain can use for fuel. Each step yields energy-rich molecules in predictable amounts, and each round shortens the fatty acid by exactly 2 carbons. The enzymes involved in the process are highly specific, recognizing fatty acids of different lengths and handling them accordingly. This precision ensures that beta oxidation unfolds smoothly, without waste or imbalance. The mitochondria, with their folded inner membranes and carefully maintained environment, provide the ideal setting for this work, an enclosed, calm chamber where energy can be extracted with the care of a practiced artisan. The regulation of beta oxidation also reflects the body's desire for balance. When carbohydrates are abundant, insulin signals the body to store fat and slow fat breakdown. When carbohydrates are scarce, hormones such as glucagon gently encourage the release of fatty acids from adipose tissue, increasing their availability for oxidation. This hormonal interplay prevents conflicting signals, ensuring that the body uses fats and carbohydrates in a coordinated way. Cells do not burn fats aggressively when glucose is plentiful, nor do they rely heavily on glucose when fats are available. Instead, the body shifts softly between fuel sources, matching energy use to energy availability. Beta oxidation not only provides energy, it also helps maintain metabolic flexibility. This flexibility allows the body to adapt to changes in diet, activity, and energy needs. It enables endurance during long periods of quiet work, supports the body during sleep, and provide stability during fasting. This adaptability is a hallmark of metabolic health, and beta oxidation plays a central role in sustaining it. As fatty acids are gradually transformed, the body carries on with its many quiet tasks, repairing tissues, maintaining body temperature, generating new molecules, and supporting calm thought, all fueled by this slow oxidation. Even when the mind is peaceful and the body rests deeply, beta oxidation continues with soft persistence, providing the ATP needed to maintain the internal harmony that keeps life moving gently forward. In the next part of this exploration, we will turn from lipids to another group of essential nutrients, proteins. Their digestion and metabolism reflect an entirely different kind of transformation, one centered not only on energy but on structure and renewal. But before stepping into that world, it is soothing to appreciate the steady rhythm of beta oxidation, the quiet unraveling of fatty acids that supports the body with calm, enduring energy. 14. Proteins, building blocks, and metabolic chameleons. Proteins enter the story of nutrition with a quiet importance that is unlike any other nutrient. While carbohydrates offer quick and accessible energy, and lipids provide long and steady reserves, proteins serve as the raw materials from which the body builds, repairs, and reshapes itself each day. They are the scaffolding of life, forming muscle fibers that contract gently, enzymes that guide delicate reactions, hormones that whisper messages between organs, and structural components that hold cells in calm harmony. They are metabolic chameleons, able to shift from one role to another depending on the body's needs, participating in countless pathways that help maintain balance, resilience, and soft continuity within every tissue. Proteins come to the body through a wide variety of foods: beans, grains, dairy, meat, fish, eggs, nuts, seeds, and even the quiet green leaves of plants. Each food offers its own arrangement of amino acids, the small molecules that form the foundation of every protein. These amino acids are linked together in long chains, curled and folded into intricate shapes that allow them to perform their functions with gentle precision. When we eat protein-containing foods, the body does not absorb them as intact structures. Instead, it patiently takes them apart, releasing the amino acids so they can be used to create new proteins unique to the body's own needs. This transformation begins in the stomach, where a warm, acidic environment helps unfold the complex shapes of dietary proteins. The acid does not break the proteins into individual amino acids, but it loosens the folded structures, making them easier for enzymes to reach. Pepsin, the primary protein-digesting enzyme in the stomach, begins its slow and steady work by breaking these long chains into shorter fragments called peptides. This work does not happen abruptly. It happens in waves of small steps, each one preparing the fragments for the next phase of digestion in the small intestine. As the partially digested proteins move into the small intestine, they encounter a new set of enzymes released by the pancreas: trypsin, chymotrypsin, elastase, and carboxypeptidases. These enzymes continue the breakdown, gently cleaving the peptide chains into smaller and smaller pieces. Each enzyme recognizes particular bonds, slowly reducing the complex structure of dietary proteins into dipeptides, tripeptides, and free amino acids. Along the brush border of the small intestine, even further breakdown occurs, as enzymes embedded in the membrane complete the process, ensuring that the body absorbs only the simplest and most usable forms. Absorption follows, a calm and organized pattern. Amino acids and small peptides cross into the enterocytes through specialized transport proteins, each one selective for different groups of amino acids. Some require active transport, using energy to move molecules against gradients, while others flow inward more gently through facilitated diffusion. Once inside the enterocytes, any remaining small peptides are broken down into individual amino acids. These amino acids then drift into the capillaries of the small intestine and enter the hepatic portal vein, beginning their journey to the liver. The liver stands at the crossroads of protein metabolism. It receives the newly absorbed amino acids and evaluates how they should be used. Some are sent out into the bloodstream to nourish muscles, organs, and tissues that require constant replenishment. Some are transformed into other amino acids that the diet may have lacked. Some become precursors for hormones, neurotransmitters, or structural proteins. Liver may even use amino acids to produce energy or to synthesize glucose when the body needs it. This versatility is one of the defining features of proteins. They can be molded into forms far beyond their dietary origin, becoming whatever the body requires at that moment. Yet the role of proteins in metabolism extends far beyond simple rebuilding. Amino acids can undergo deamination, a gentle removal of their nitrogen-containing groups. This process allows the remaining carbon skeletons to enter metabolic pathways such as glycolysis or the citric acid cycle. Some amino acids become pyruvate, others become acetyl-CoA. Still others transform into citric acid cycle intermediates. Through these pathways, proteins can contribute to energy production, especially during times when carbohydrate intake is low or when the body's energy needs exceed the supply. This ability to switch roles to serve as structure, message, repair material, or energy source makes proteins truly adaptable, earning them the title of metabolic chameleons. The nitrogen removed during deamination cannot remain in the body freely, as it could disrupt the delicate balance of the internal environment. Instead, the liver converts it into urea, a harmless compound that the kidneys later remove through urine. Even this process, known as the urea cycle, is steady and calm, ensuring that nitrogen is released from amino acids without disturbing cellular harmony. It reflects the body's care in handling proteins, using them fully but always maintaining balance. Proteins also play key roles in the body's immune system. Antibodies proteins with graceful, branching arms recognize and neutralize foreign substances. Their ability to bind with specificity is one of the body's most intricate defenses, built from amino acids arranged in patterns that allow them to identify even the smallest molecular details. Enzymes, another class of proteins, pathways with a gentle touch that lowers barriers and encourages bonds to shift. Without enzymes, reactions essential to life would occur far too slowly. With them, metabolism unfolds with quiet elegance. Hormones, such as insulin and glucagon, are also proteins. Insulin guides glucose into cells, while glucagon releases stored glucose when levels begin to fall. These hormones ensure that the relationship between carbohydrates, fats, and proteins remains coordinated. Their presence shows that proteins are deeply embedded in every aspect of metabolism, not only building the machinery of cells, but directing the flow of nutrients moment by moment. Inside muscles, proteins take on a different role. They form the fibers that contract and relax, enabling movement, posture, and subtle shifts in expression. These proteins, arranged in overlapping filaments, slide past one another in a smooth, continuous pattern that creates motion. When muscles are used, they require constant repair and renewal. Even at rest, muscles undergo quiet cycles of repair, and proteins supply the material that makes this possible. In the brain, proteins form the receptors that receive neurotransmitters, the channels that allow ions to flow, and the enzymes that maintain electrical balance. The thoughts we think, the memories we form, and the calm we experience all depend on protein structures that guide neural activity. These proteins change and adapt in response to experience, illustrating once again the flexibility that defines them. Even at the cellular level, proteins support life by forming the cytoskeleton, the internal framework that holds each cell in shape. These filaments strengthen the cell against external pressures, anchor organelles, and help transport molecules from one region to another. They move slowly and steadily, supporting the inner architecture with quiet persistence. What makes proteins especially remarkable is how tightly they are woven into every part of the body's function. Unlike carbohydrates and fats, which often serve specific and focused roles, proteins participate in nearly everything. They do not merely sustain life, they shape and direct it. Through their ability to fold into endless forms, to catalyze countless reactions, and to shift into different pathways depending on need, proteins embody the adaptability and quiet wisdom of biochemical life. As we move toward the next part of this journey, the focus will turn to how amino acids, once freed and absorbed, join the deeper pathways of metabolism, entering energy cycles, or becoming intermediates that support the body's dynamic needs. But for now, it is enough to rest with the understanding that proteins are the body's gentle architects and versatile workers, forming the soft foundation on which every structure and process depends. 15. Protein digestion, from stomach to small intestine. Protein digestion is one of the most intricate and gentle transformations within the body, an unfolding that mirrors the slow rhythm of growth, repair, and renewal. Unlike carbohydrates, which dissolve quickly under the influence of enzymes, or fats, which drift through the digestive system in calm droplets, proteins require a more layered and patient approach. Their structures are complex, folded into delicate shapes that govern how they function in the bodies from which they came. When we consume proteins, we are inviting these intricate molecules into a long, quiet journey of unfolding, fragmenting, and rebuilding. It is a journey that begins in the warm depths of the stomach and continues through the soft corridors of the small intestine, eventually yielding the amino acids that the body will use to create its own unique proteins. The process begins even before food reaches the stomach. When the senses detect the presence of a meal, the sight, smell, or even memory of food, the body prepares itself through gentle, anticipatory signals. Gastric glands begin secreting gastric juices, and the stomach's muscular layers relax slightly, ready to receive nourishment. Once protein-containing foods enter the stomach, they encounter a world very different from the one outside. A warm, acidic environment designed to soften and unfold the complex structures proteins naturally hold. The stomach's acidity is maintained primarily by hydrochloric acid, which lowers the pH and creates ideal conditions for the first major step of protein digestion. This acidic environment does not immediately break proteins into amino acids. Instead, it begins the process with a quiet unfolding of their three-dimensional shapes. Proteins, which often curl into elaborate folds to perform their functions, begin to relax and open under these acidic conditions. Their bonds loosen, allowing enzymes to access the tightly held chains of amino acids. This unfolding is called denaturation, and although the word may sound harsh, the process itself is gentle, gradual, and essential for proper digestion. Into this softened environment enters pepsin, the main proteolytic enzyme of the stomach. Pepsin is released initially as an inactive form called pepsinogen, a safeguard that prevents it from acting prematurely. Only when pepsinogen meets the stomach's acidity does it transform into active pepsin, ready to begin its slow and steady work. Pepsin does not break protein chains entirely into free amino acids. Instead, it cleaves the long chains into shorter fragments called peptides. These peptides still hold the essence of their original structure, but they have been reduced to simpler forms that the small intestine can handle with ease. Throughout this process, the stomach's muscular walls perform gentle waves of contraction, mixing the food with gastric juices to ensure complete and even digestion. These waves, known as peristaltic movements, are rhythmic and calm, helping to transform the mixture of food and digestive fluids into chyme, a smooth, semi-liquid substance. Chyme allows nutrients to move more easily through the intestinal tract, contributing to a digestion process that unfolds with natural pacing rather than urgency. As the chyme reaches a certain consistency, The stomach slowly releases it through the pyloric sphincter into the duodenum, the first section of the small intestine. This release occurs in measured amounts, ensuring that the small intestine is never overwhelmed and can continue its work peacefully. The entrance of chyme into the duodenum triggers hormonal signals, chief among them secretin and cholecystokinin, which prompt the pancreas to release its digestive juices. These pancreatic secretions hold a new family of protein-digesting enzymes, each with its own subtle role. Trypsin, chymotrypsin, elastase, and carboxypeptidases are among the main proteases released by the peptidases of the duodenous pancreas. Like pepsin, many of these enzymes are secreted in inactive forms, known as zymogens. This protective mechanism ensures they do not begin digesting proteins too early or in the wrong environment. Once in the duodenum, these zymogens encounter activating enzymes. Trypsinogen becomes trypsin, which then activates the remaining proteases. This cascade of activation mirrors a gentle chain reaction, building momentum just enough to support digestion without disturbing the structural harmony of the surrounding tissues. In the small intestine, the environment is no longer acidic. Pancreatic bicarbonate neutralizes the gastric acid, creating a slightly alkaline space that supports the activity of intestinal enzymes. Here, the peptides, once created by pepsin, are broken down further into smaller fragments: dipeptides, tripeptides, and amino acids. Each enzyme has a preference for certain peptide bonds, working on its selected targets in a manner that ensures thorough but gradual digestion. The brush border of the small intestine, lined with microvilli, contributes additional enzymes that complete the breakdown. These brush border enzymes, embedded directly in the membrane of the enterocytes, quietly perform the final steps, ensuring that the products of digestion are small enough for absorption. The absorption of amino acids and small peptides is a gentle and finely tuned process. Transport proteins in the membranes of enterocytes recognize different types of amino acids, neutral, acidic, basic, or sulfur-containing, and usher them across with precision. Some of these transports require energy, while others are facilitated by gradients that guide the nutrients inward. Small peptides that enter the enterocytes are broken down into individual amino acids, ensuring that the bloodstream receives only the simplest, most usable forms. Once the amino acids enter the bloodstream, they travel through the hepatic portal system, reaching the liver first. The liver examines this influx of amino acids and decides how best to distribute them. Some will be sent to tissues immediately, supporting growth, repair, enzyme synthesis, and hormonal signaling. Others will be stored temporarily or converted into different amino acids the body may be lacking. In moments when energy is needed, some amino acids may even be directed into pathways that allow them to contribute fuel to the system. The careful unfolding of protein digestion reflects the body's deep respect for these molecules. Proteins represent not just nutrients, but tiny structures crafted with intention by the organisms from which they came. Their amino acids served other lives before becoming part of yours, and their breakdown is performed with the same quiet dignity that characterizes all of digestion. The stomach's warm acid opens their folds. The intestine's calm enzymes reshape them. The liver gently repurposes them for new needs. Within the cells of the body, these amino acids will eventually become new proteins, muscle fibers, enzymes, transporters, antibodies, or structural frameworks. They will be woven into the architecture of life, becoming part of thoughts, movement, healing, and resilience. As we move forward into the next part of this calming exploration, we will follow these amino acids further, tracing how they enter metabolic pathways and transform into intermediates that support energy production and physiological balance. But for now, the peaceful process of protein digestion offers a reminder that nourishment is always a patient unfolding, a slow and mindful transition from the foods we eat into the substance of our very being. 16. Amino acid pathways and entry into central metabolism. Once proteins have been gently broken down into their individual amino acids and carried softly through the bloodstream, they arrive at the crossroads where the body decides how each one will be used. This moment is quiet, yet profound. Every amino acid becomes a small participant in a much larger story, a story that includes growth, repair, energy, and the seamless integration of nutrients into the central pathways that sustain life. Unlike carbohydrates, which follow clear and predictable routes, or lipids, which offer slow and enduring energy, amino acids possess a unique flexibility. They can transform into structures, signals, enzymes, or fuels. They can build, restore, or energize. This adaptability makes them essential not only for the body's architecture, but also for the fluid harmony of metabolism. After amino acids reach the liver through the portal vein, they find themselves in one of the most metabolic tissues in the body. The liver examines and sorts them, much like a careful guardian tending to a diverse collection of delicate materials. Some amino acids are released almost immediately into the bloodstream so that muscles, organs, and tissues can use them. Others are converted into slightly different amino acids, ensuring that the body has a balanced supply. Still others are held temporarily in the liver's quiet reserve, available for moments when the tissues require them. This distribution reflects a deep wisdom within the body. Amino acids should never be wasted, and their use must align with the shifting needs of each moment. When amino acids are used for building new proteins, enzymes, receptors, hormones, and structural components, the body places them carefully into long chains, folds these chains into specific shapes, and guides them toward their appropriate roles. But when the body requires energy, or when certain amino acids are in excess, they are gently guided into metabolic pathways. These transformations begin with deamination, the removal of the amino group. Instead, it unfolds through the patient work of enzymes that release the nitrogen-containing component so that the carbon skeleton can be used for energy. The nitrogen, once removed, is carried into the urea cycle, where it is converted into urea, a safe, soluble form that the kidneys can eliminate quietly. The urea cycle itself is a peaceful turning of reactions within the liver, functioning steadily to maintain balance. Once the amino group has been lifted away, the remaining carbon skeleton can slip into one of several metabolic pathways. Some amino acids are transformed into pyruvate, a well-known intermediary that serves as a gateway to both aerobic and anaerobic metabolism. These amino acids are called glucogenic because through pyruvate they can support the creation of glucose, a gentle process that becomes essential during fasting or times of low carbohydrate intake. Pyruvate can also enter the mitochondria, becoming acetyl-CoA, or feeding into the citric acid cycle. In this way, glucogenic amino acids enter metabolism through familiar doors, joining pathways already prepared to welcome them. Other amino acids become acetyl-CoA directly. These are called ketogenic amino acids, and they support metabolic flexibility in moments when glucose is scarce. Acetyl-CoA, derived from these amino acids, enters the citric acid cycle, or contributes to the formation of ketone bodies in the liver. Ketone bodies serve as a calm, alternative energy source during prolonged fasting, low carbohydrate diets, or extended periods of rest between meals. They drift through the bloodstream gently, offering fuel to the brain, heart, and other tissues when glucose levels fall. Amino acids that support this pathway help expand the body's ability to maintain balance across different conditions. Some amino acids enter the citric acid cycle directly without becoming pyruvate or acetyl-CoA first. These amino acids slip into the cycle at different points, sometimes as alpha-ketoglutarate, sometimes as succinyl-CoA, fumarate, or oxaloacetate. By entering at these various stages, they nourish the cycle, replenishing intermediates that may have been used elsewhere for biosynthesis. This replenishment is known as anaplerosis, and it allows the citric acid cycle to continue its smooth turning even when some of its molecules are drawn away for other tasks. Without amino acids contributing in this way, the cycle would slow or falter, disrupting the peaceful continuity of energy production. The flexibility of amino acid metabolism reflects the body's ability to adapt. During periods of abundant food intake, amino acids participate primarily in building, supporting muscle maintenance, enzyme production, and tissue repair. During fasting, they help maintain blood glucose levels and supply substrates for the citric acid cycle. During prolonged exertion, they step in to support energy production when glycogen stores begin to fall. This adaptability ensures that no matter the circumstance, the body retains the ability to sustain itself without abrupt changes or instability. Amino acids also feed into pathways that produce neurotransmitters, the quiet chemical, messengers of the nervous system. Tryptophan becomes serotonin, a molecule associated with mood, sleep, and calm. Tyrosine becomes dopamine and norepinephrine, supporting focus and alertness. Glutamate becomes GABA, the primary inhibitory neurotransmitter that helps quiet the mind and promote relaxation. These conversions show that amino acid metabolism extends beyond energy and structure. It touches on thought, emotion, and the gentle rhythms of the nervous system. Some amino acids support the synthesis of molecules such as creatine, which helps muscles store energy for short bursts of activity. Others contribute to the creation of nucleotides, the building blocks of DNA and RNA, allowing cells to divide and repair themselves. Still others become components of antioxidants, helping protect cells from stress. In all these roles, amino acids demonstrate their extraordinary versatility. Even when amino acids enter metabolism for energy, their use is never abrupt or careless. The body prefers to use carbohydrates and fats first, conserving amino acids for structure and function. Only when needed do amino acids become fuel, reflecting a deep instinct for preservation. This priority ensures that muscle tissue remains intact and that essential proteins continue to function. As amino acids move through these pathways, they join the continuous flow of metabolism, a river of reactions that supports warmth, breath, thought, and quiet life. Their transformations are soft, reversible when possible. Whether becoming glucose during fasting, entering the citric acid cycle during movement, or contributing to neurotransmitter synthesis during rest, amino acids play their roles with subtlety and grace. As we move forward to the next part of this exploration, we will examine how the body chooses which fuel to use at any moment, and how carbohydrates, fats, and proteins quietly collaborate to maintain balance. For now, it is enough to rest in the understanding that amino acids are not merely nutrients, they are the adaptable threads that weave metabolism into a coherent whole, supporting life in every form it takes. 17. Metabolic integration of carbohydrates, fats, and proteins. Within the body, metabolism does not operate as a series of isolated pathways. Instead, it unfolds as a gentle, interconnected network where carbohydrates, fats, and proteins share a quiet, ongoing conversation. Each nutrient follows its own path, shaped by its structure and purpose. But these paths never remain separate for long. They braid together softly at many points, meeting in shared intermediates, overlapping in cycles, and adjusting to one another in response to the body's changing needs. This integration ensures that energy flows steadily, that tissues receive the right fuel at the right moment, and that life continues in a state of balance, calm, adaptable, and deeply coordinated. Carbohydrates enter metabolism quickly. When glucose enters the cell, it passes through glycolysis, forming pyruvate, which then transitions into acetyl-CoA. This molecule represents one of the central meeting points of metabolism, the peaceful crossroads where all three nutrient types eventually gather. Acetyl-CoA enters the citric acid cycle, feeding a sequence of reactions that gently extract energy in the form of NADH, FADH2, and ATP. This cycle sits at the center of cellular metabolism, welcoming not only carbohydrates but also fatty acids and certain amino acids. Because of this convergence, the citric acid cycle acts as a shared rhythm of life, its soft circular motion connecting the different sources of energy the body may call upon. Fats enter metabolism more slowly. Before entering the citric acid cycle, long-chain fatty acids undergo beta oxidation, a stepwise process that trims them into two carbon segments of acetyl-CoA. These fragments slip quietly into the same pathway used by carbohydrates. There is no distinction within the cycle between an acetyl-CoA that once came from a grain of rice, one that came from a drop of olive oil, or one released from a resting muscle's triglyceride stores. The cycle treats them equally, illustrating the unity of metabolic design. Proteins join this interconnected network in yet another way. After amino acids undergo deamination, their carbon skeletons are shaped into intermediates that enter glycolysis, or the citric acid cycle. Some become pyruvate, others become acetyl-CoA. Still others turn into intermediates such as fumarate, succinyl-CoA, or oxaloacetate. Each one merges smoothly into the pathways already turning, contributing to energy production, replenishing intermediates, or supporting biosynthesis. This ability to join metabolism at many points makes proteins particularly adaptable, providing support where it is needed most. Throughout these pathways, intermediates shuttle back and forth in quiet cycles. Pyruvate can become acetyl-CoA, but, under different circumstances, it may also support gluconeogenesis, becoming glucose again when blood sugar falls. Oxaloacetate can help drive the citric acid cycle, or it can leave the cycle to support glucose formation. Acetyl-CoA can enter the cycle for energy, or, in low carbohydrate conditions, it can shift toward the production of ketone bodies, offering gentle support to the brain and muscles when glucose is scarce. These flexible transitions create metabolic resilience, allowing the body to adapt to fasting, feeding, movement, or rest. Insulin, released after meals, signals the body to use and store nutrients. It also supports protein synthesis by guiding amino acids into cells. When insulin is present, the pathways of storage gently open. When glucose falls, glucagon steps in to guide the body through a different metabolic landscape. It encourages the release of stored glucose, the mobilization of fatty acids, and the gentle shift of amino acids into gluconeogenesis. Cortisol, growth hormone, epinephrine, and thyroid hormones add further layers of guidance, weaving a hormonal tapestry that ensures metabolism responds not only to food but also to temperature, activity, time of day, and emotional state. Because of this hormonal interplay, the body rarely depends on a single nutrient at any moment. During the calm hours after a meal, carbohydrates may be the primary fuel, while fats quietly enter storage. During fasting or gentle overnight sleep, fats provide the slow, steady energy that maintains breathing, temperature, and heartbeat. During longer periods without food, proteins contribute their carbon skeletons to maintain blood glucose, ensuring that the brain continues to function smoothly. Even within a single cell, different parts may use different fuels, depending on their needs. The liver orchestrates energy distribution. Muscles draw upon glycogen during brief activity, switch to glucose during moderate exertion, and rely heavily on fatty acids during longer, quieter motion. The heart prefers fatty acids almost all the time, savoring their slow and steady energy. The brain relies on glucose but can shift to ketone bodies when necessary. The citric acid cycle, sitting at the center of this network, reflects the unity of metabolism. Its intermediates rise and fall in response to the availability of nutrients. When carbohydrates are abundant, oxaloacetate increases to support the processing of acetyl-CoA. When fats dominate, the cycle adjusts, allowing acetyl-CoA to enter at a pace matched by available intermediates. When amino acids contribute intermediates, the cycle stabilizes itself, strengthening its rhythmic turn. Through these adjustments, the cycle maintains steady energy production even as nutrient sources shift. Another gentle point of integration is the way excess nutrients flow into storage. Excess glucose becomes glycogen or, when stores are full, becomes fatty acids. Excess fatty acids become triglycerides, settling into adipose tissue with patience. Excess amino acids cannot be stored directly, so their carbon skeletons move into metabolic pathways, becoming glucose or fat when necessary. This redistribution ensures that nothing is wasted and everything finds its place. When nutrients are scarce, the process reverses. Glycogen slowly releases glucose. Triglycerides become fatty acids. Protein reserves, though protected as much as possible, contribute when essential. The body's design supports harmony, allowing the transitions between these states to feel seamless and calm. The interconnectedness of metabolism becomes particularly apparent during exercise. In the first moments of movement, muscles rely on stored ATP and phosphocreatine. As activity continues, glycolysis supplies energy. With longer activity, the citric acid cycle and oxidative phosphorylation take over. Amino acids contribute only slightly under normal circumstances, yet their presence ensures that metabolism remains steady. This shifting reliance shows how the body integrates all nutrient pathways to support continuous, smooth function. Even at rest, metabolic integration plays a quiet role in maintaining life. The brain, heart, and kidneys require steady energy, and the body provides it by blending fuels as needed. During sleep, fats often take the lead, supported by small contributions from glucose and occasional help from amino acids. This nighttime balance protects blood sugar and ensures that tissues receive exactly what they need without strain. As the pathways of carbohydrates, fats, and proteins intertwine, they create a flexible and peaceful network. The body does not treat these nutrients as separate categories, but as partners in the shared work of sustaining life. Their integration reflects the underlying unity of metabolism, where every molecule has a place, every pathway has a purpose, and every shift unfolds with quiet precision. In the next part of this journey, we will turn toward the body's elegant storage systems, how nutrients are tucked away in glycogen granules, adipose droplets, and amino acid pools, waiting patiently until the next moment they are needed. For now, it is enough to rest with the understanding that metabolism is not a collection of isolated reactions, but a gentle, continuous web where all nutrients meet, cooperate, and support one another. 18. Energy storage, balancing immediate needs and future reserves. Within the quiet chemistry of the body, one of the most remarkable expressions of balance is its ability to store energy. Life does not unfold in perfectly timed intervals of nourishment. There are moments of abundance and moments of scarcity, moments when food arrives easily, and moments when the body must rely on its own reserves. To remain steady through all of these fluctuations, the body has developed gentle systems for setting aside energy, preserving it in a safe and stable form, and releasing it again with soft precision when needed. These systems for storing carbohydrates, fats, and proteins operate continuously and quietly, ensuring that energy is available whether hours, days, or even weeks have passed since the last meal. Carbohydrates are stored primarily as glycogen, a branching chain of glucose molecules. Glycogen is not scattered randomly through the body. It forms small, organized granules in the liver and muscles, tucked away like tiny reservoirs that can be drawn upon at any moment. In the liver, glycogen serves as a calm guardian, of blood glucose levels. When blood sugar begins to fall, whether between meals, during sleep, or during a long period of focus, the liver gently breaks down its glycogen reserves, releasing glucose into the bloodstream. This action keeps the brain supplied with fuel, maintains body temperature, and supports the quiet functioning of vital organs. Muscle glycogen serves a different purpose. It does not leave the muscle. Instead, it provides a local supply of energy, supporting movement, posture, and the subtle contractions that maintain balance and stability. The process of glycogen storage begins in the fed state, guided by the hormone insulin. After a meal, when glucose flows through the bloodstream, insulin encourages cells to absorb it. In the liver, insulin activates enzymes that link glucose molecules together into glycogen. The branching structure of glycogen allows it to hold a large amount of glucose in a compact form, and the many branches enable rapid release when needed. Glycogen synthesis is not hurried. It unfolds in soft waves, matching the pace of nutrient availability. When reserves become full, excess glucose gently shifts toward other pathways, eventually contributing to fat synthesis if energy intake continues to exceed immediate needs. Fats represent the body's most concentrated and enduring form of stored energy. Unlike glycogen, which holds water and is limited in capacity, fats are stored in an anhydrous form, allowing vast amounts of energy to be preserved without occupying much space. Adipose tissue, found beneath the skin and around internal organs, serves as the primary site of fat storage. Within each adipocyte, or fat cell, triglycerides form small droplets that expand slowly as new fat is added. These droplets remain soft and pliable, held in suspension by a delicate framework of proteins. Fat storage is guided by a rhythm that responds to the body's internal signals. When insulin is present after eating, or during moments of calm and nourishment, the body encourages the storage of fatty acids. These fatty acids come from dietary fats carried by chylomicrons, or they are produced in the liver and transported in the form of very low-density lipoproteins. Inside adipocytes, the fatty acids are reassembled into triglycerides and tucked away for the future. This process is not rigid. It allows for steady ebb and flow, adding fat when energy is plentiful. When food intake decreases or energy demands rise, triglycerides within adipose tissue break down through lipolysis. Hormones such as glucagon and epinephrine signal the fat cells to release fatty acids into the bloodstream, where they bind to albumin and travel softly to tissues that can oxidize them for energy. This gentle release helps maintain energy availability during fasting, extended sleep, or quiet endurance activity. Because fats contain so much stored energy, they are capable of supporting the body for long periods without causing strain or rapid depletion. Proteins, unlike carbohydrates and fats, are not stored in dedicated reserves. The body values them too highly as functional molecules, muscle fibers, enzymes, hormones, and structural frameworks to set them aside for later use. Instead, the body maintains a circulating pool of amino acids created from dietary intake, ongoing protein turnover, and the gentle recycling of cellular components. These amino acids move softly between tissues, always available for new protein synthesis or, when necessary, for energy production. The lack of a storage system for proteins means that the body relies heavily on carbohydrates and fats to meet energy needs, preserving proteins for their essential roles. Only when energy becomes scarce for extended periods do amino acids begin to shift more noticeably toward energy production, supporting gluconeogenesis or entering the citric acid cycle as intermediates. The harmony of energy storage depends on the interplay of hormones. Storage after meals. When insulin levels rise, glycogen synthesis increases, fatty acid deposition is encouraged, and protein synthesis unfolds smoothly. During fasting, insulin levels fall. Epinephrine supports similar processes during moments of activity or stress. Cortisol, Though often thought of as a stress hormone, quietly supports gluconeogenesis and helps maintain blood glucose levels during prolonged fasting. Through these hormonal shifts, the body moves seamlessly between storing energy and releasing it, maintaining balance without dramatic changes. The timing of nutrient storage reflects the natural rhythms of life. After a meal, nutrients flow towards storage. During sleep, when the body rests and digestion slows, stored energy maintains the quiet processes of repair and renewal. During moments of movement, fatty acids and glycogen supply energy in proportion to the intensity and duration of activity. If the body experiences extended periods without food, it shifts gently into a fasting state, relying first on glycogen, then on fats, and finally, if needed, drawing on amino acids to sustain vital functions. These transitions unfold without abrupt shifts, guided by the body's innate desire for stability. The integration of storage systems also protects the body from extremes. If glycogen were the only form of stored energy, it would be depleted quickly, leaving the body vulnerable. If fats were the only form, the body would lack a rapid source of glucose, especially for the brain. If proteins were used constantly for energy, muscle and organ function would be compromised. By maintaining a blend of storage systems, quick access glycogen, long-lasting fats, and flexible amino acid pools, the body preserves its structural integrity and functional harmony. Within cells, energy storage takes place in even subtler forms. ATP itself is not stored in large quantities but is regenerated continuously. Phosphocreatine, found in muscles, provides a small, immediate reserve for sudden needs. Glycogen granules sit quietly among the fibers of muscle cells, ready to fuel contraction. Triglyceride droplets in muscle tissue offer additional energy during longer periods of activity. This layering of energy reserves ensures that the body can meet both sudden demands and slow, sustained needs without disruption. As we move toward the next part of this journey, we will explore how nutrients flow not only in cycles of storage and release, but also in cycles of waste removal. How the body eliminates byproducts gently and efficiently, maintaining internal clarity. For now, it is enough to appreciate the elegance of energy storage, the quiet readiness of glycogen, the patient depth of fat deposits, and the adaptable nature of amino acid pools. These systems together form a peaceful foundation that sustains life through its many rhythms. 19. Metabolic waste and the gentle art of cleansing. As nutrients flow through the body and are transformed into energy, building blocks, and essential intermediates, they leave behind small traces, byproducts of metabolism that must be guided out of the body with care. These remnants are not harmful in themselves, but if allowed to accumulate, they could disturb the delicate balance that metabolism strives to maintain. The body therefore practices a quiet art of cleansing, a continuous, unobtrusive process that removes metabolic waste as naturally as breathing removes carbon dioxide. This process unfolds in soft, rhythmic layers, handled by organs that filter, neutralize, package, and release these materials with patient precision. Far from being abrupt or aggressive, metabolic waste removal is one of the body's most calming and steady functions. Among the first and most constant byproducts of metabolism is carbon dioxide. Every cell produces this gentle gas as it turns nutrients into energy through the citric acid cycle and oxidative phosphorylation. Carbon dioxide diffuses out of cells into the bloodstream, where it dissolves into plasma or binds softly to hemoglobin. Carried by the circulatory system, it travels to the lungs. There, with each slow and quiet exhale, carbon dioxide is released into the air. Breathing, though often unnoticed, is the body's most continuous expression of waste removal. Each breath reflects a perfect balance. Oxygen enters to support cellular life, and carbon dioxide leaves as a peaceful sign that metabolism is unfolding as it should. This exchange helps regulate the body's pH, ensuring the internal environment remains stable and serene. When amino acids are used for energy or transformed into intermediates, their nitrogen-containing groups are gently removed through deamination. Free ammonia, the initial form of this nitrogen, is highly reactive and must be handled with care. The liver takes responsibility for this task, guiding ammonia through the urea cycle. In this cycle, ammonia is combined with carbon dioxide and other intermediates to produce urea, a neutral and safe compound. The urea cycle's turning is quiet and continuous, protecting the body from any disruption while converting nitrogen into a form that can move peacefully through the bloodstream. Urea travels to the kidneys, two organs that serve as the body's primary filters. Each kidney contains millions of nephrons, tiny filtering units that work gently and tirelessly. Blood flows through these nephrons, and waste substances like urea, creatinine, and small amounts of other byproducts drift into the forming urine. The kidneys are not merely filters, they are regulators, adjusting water balance, electrolytes, and acid-base levels with subtle precision. When the body is well hydrated, urine becomes lighter and more diluted. When water is scarce, the kidneys concentrate urine, conserving water while still eliminating waste. Throughout these shifts, the process remains smooth, reflecting the kidney's role in maintaining internal calm. Creatinine is another quiet byproduct, formed in the muscles from the breakdown of creatine phosphate, a molecule involved in quick energy release. Creatinine production remains steady, flowing from muscles into the bloodstream and then into the kidneys for elimination. Because its rate of formation is stable, creatinine becomes a gentle indicator of kidney function, reflecting how well this filtration system is working without requiring any conscious attention. The liver plays a central role not only in protein waste processing but in the detoxification of various metabolic intermediates. It receives blood rich in nutrients and byproducts from the digestive tract and performs a series of transformations that neutralize potentially reactive molecules. Alcohol, medications, and natural metabolic byproducts all pass through the liver, where enzymes modify them into forms that can be released safely. These processes, often described as phases of detoxification, involve oxidation, conjugation, and preparation for excretion. Each step is slow and deliberate, ensuring that substances are rendered harmless before they enter the bloodstream or move toward elimination. Bile, another pathway for waste removal, is produced by the liver and stored in the gallbladder. It carries not only bile salts for fat digestion but also waste products like bilirubin. Hemoglobin from aging red blood cells is dismantled, its components reused or removed. Bilirubin travels to the liver, where it is conjugated, made water-soluble, and then excreted into bile. When bile enters the small intestine during digestion, bilirubin travels with it, eventually leaving the body through the stool. This gentle recycling of red blood cells ensures that vibrant, new cells can take their place while the byproducts of their breakdown exit the body in a calm, steady flow. Water itself plays a significant role in cleansing. Through sweat, the body releases small amounts of metabolic waste, water, urea, salts, and trace substances. Sweating is not primarily a waste removal system, but its contribution is meaningful. It helps maintain temperature balance, allowing the body to cool itself while simultaneously eliminating minor waste products. The skin, through its pores, becomes part of the body's broader network of quiet purification. The lymphatic system also participates in waste movement, but in a more subtle way. Lymph collects fluid that has seeped into tissues, carrying with it small molecules, cell debris, and immune components. This fluid moves slowly through lymphatic vessels, propelled mainly by muscle movement and natural body rhythms. Eventually, it reaches lymph nodes, small stations where immune cells gently remove unwanted materials. Cleaned lymph then returns to the bloodstream, ready to continue circulating. Within cells, there are equally gentle systems for waste management. Lysosomes act as small recycling centers, breaking down worn-out organelles, misfolded proteins, and cellular debris. They digest these materials into basic components that the cell can reuse or eliminate. This internal housekeeping ensures that the cell remains clear, functional, and calm. Autophagy, a related process, allows cells to surround and dismantle their own damaged parts during times of scarcity or stress. Rather than being destructive, autophagy is a serene form of self-renewal, an internal cleansing that supports long life and resilience. The removal of free radicals, reactive molecules generated during normal metabolism, is handled by antioxidants and enzymatic pathways. These systems work continuously to neutralize reactive oxygen species, preventing them from disturbing cellular peace. The body produces its own antioxidants, such as glutathione, superoxide dismutase, and catalase, which quietly maintain balance, while nutrients like vitamin C and vitamin E supplement this protection. This quiet shield ensures that oxidative stress remains low, supporting long-term health. What makes the body's waste removal systems remarkable is not only their efficiency but their softness. They operate in cycles, responding to the body's rhythms of eating, fasting, activity, and rest. They adjust fluid levels, pH balance, and solute concentrations without requiring conscious command. They maintain the clarity of the internal environment with gentle consistency. As energy flows through the body from nutrients absorbed to ATP produced to waste released, the process resembles a flowing river, calm in its course, patient in its timing, always moving but never rushing. The removal of metabolic waste ensures that this river remains clear, supporting every cell and every system with quiet steadiness. In the next part of this journey, we will explore how all these pathways come together to maintain homeostasis, the gentle equilibrium that allows the body to remain stable through change. For now, it is enough to appreciate how gracefully the body manages the byproducts of life, guiding them outward with care, allowing every cycle of metabolism to finish with clarity and calm. 20. Homeostasis, the calm coordination of nutrient supply and demand. At the heart of the body's metabolic symphony lies a concept that is both simple and profound: homeostasis. This gentle word refers to the inner steadiness that allows the body to function smoothly despite the shifting world around it. Homeostasis is not rigid or unmoving. It is a flexible, dynamic balance, one that adjusts constantly to keep temperature, blood sugar, hydration, pH, and countless other factors within ranges that support life. The biochemistry of nutrition, with all its pathways and transformations, feeds directly into this delicate equilibrium. Every molecule absorbed, stored, used, or released becomes part of a serene, ongoing dialogue between supply and demand. Nutrients enter the body in cycles, arriving as meals spaced across the day or in smaller quantities through snacks or beverages. They do not arrive continuously, yet the body's needs are constant and unbroken. Between meals, metabolism cannot pause, tissues still require fuel, and cells still perform their quiet work. To bridge this gap, the body relies on the reserves and pathways it maintains, with such care glycogen in the liver to maintain blood sugar, fatty acids stored in adipose tissue for calm and steady energy, and amino acids circulating gently for protein synthesis or emergency fuel. These stores do not simply exist, they respond, ebbing and flowing in subtle ways that keep the body stable. When nutrients are plentiful, energy is stored. When nutrients are scarce, energy is released. The shift between these states unfolds with natural pacing. Blood glucose is one of the most finely controlled elements of homeostasis. After a meal, glucose rises softly as carbohydrates are digested and absorbed. The pancreas senses this increase and releases insulin, a hormone that moves like a gentle guide, encouraging cells to take in glucose. Muscles store it as glycogen. The liver shifts toward synthesis. Adipose tissue stores the excess as fat. Through these actions, insulin prevents glucose from rising too sharply, protecting the internal environment from sudden change. As glucose levels fall between meals or during sleep, insulin retreats, and glucagon steps forward. Glucagon whispers to the liver, encouraging it to release glucose from its glycogen stores. This soft balancing motion ensures that glucose levels do not drop too low, allowing the brain and other sensitive tissues to remain nourished. Fat metabolism also plays a central role in homeostasis, especially during periods of quiet fasting. Decreases or blood glucose begins to dip. Fatty acids flow gently from adipose stores into the bloodstream. They drift to the liver and other tissues, where they undergo beta oxidation and fuel ATP production. The shift is gradual, reflecting the body's preference for stability rather than abrupt change. If fasting continues, the liver begins producing ketone bodies from acetyl-CoA. These molecules circulate calmly and offer an alternative fuel for the brain and muscles. Ketone bodies represent one of the body's most elegant adaptations, ensuring that even when carbohydrate intake falls significantly, the mind remains clear and the tissues remain energized. Homeostasis thrives not on uniformity, but on adaptability. Protein metabolism contributes to this harmony in its own way. Although the body avoids using proteins as a primary fuel source, amino acids can support blood glucose levels through gluconeogenesis when the need arises. When nutrients are abundant, amino acids flow toward repair, growth, enzyme production, and the formation of hormones. When nutrients are low, the amino acid pool adjusts, providing material for essential processes while preserving muscle tissue as much as possible. The liver orchestrates these shifts, monitoring amino acid concentrations and deciding how to use each molecule. Through these quiet adjustments, the body maintains structure and energy without disturbing equilibrium. Water balance is another subtle but vital part of homeostasis. The body is mostly water, and the movement of this water between cells, tissues, and organs must be maintained with exceptional care. Hormones such as antidiuretic hormone, ADH, guide the kidneys to retain water when needed, or release it when hydration increases. Aldosterone adjusts sodium balance, which in turn influences water distribution throughout the body. Even the simple act of thirst arises as a gentle reminder that hydration is part of the body's steadying process. These systems respond continuously to tiny changes in fluid levels, ensuring that cells remain plump, blood pressure stays stable, and metabolic reactions occur in the right environment. Temperature regulation is another peaceful example of homeostasis. The body maintains a stable internal temperature through mechanisms that are both subtle and efficient. When the environment grows cool, the body increases heat production through shivering, constricts blood vessels near the skin to reduce heat loss, and encourages behaviors such as seeking warmth. When warmth increases, the body releases heat by dilating blood vessels and producing sweat. The evaporation of sweat cools the skin and brings temperature back into balance. These temperature-related shifts occur with a calm rhythm, supporting the enzymes and reactions that depend on a narrow temperature range. The buffering systems of the body also support homeostasis by regulating pH. Metabolism produces acids, carbonic acid from carbon dioxide, lactic acid from anaerobic glycolysis, and acidic intermediates from protein metabolism. These acids are neutralized through systems involving bicarbonate, hemoglobin, phosphate, and plasma proteins. The lungs assist by removing carbon dioxide, preventing its accumulation. The kidneys fine-tune pH by excreting hydrogen ions and reabsorbing bicarbonate. Through these quiet exchanges, the body keeps its internal pH within a narrow window, protecting the integrity of cells and enzymes. Homeostasis also extends to micronutrients such as iron, calcium, magnesium, and potassium. These minerals participate in essential functions: oxygen transport, muscle contraction, nerve conduction, and enzyme activity, and their levels must remain balanced. The body absorbs what it needs, stores small amounts, and eliminates excess gently. Hormones such as parathyroid hormone and calcitonin guide calcium balance. Aldosterone helps regulate electrolytes, and transport proteins, shuttle minerals, where they're needed. Even the smallest fluctuations are monitored and corrected with soothing precision. Underlying all these mechanisms is a soft interplay between the nervous and endocrine systems. The hypothalamus, deep within the brain, acts as a quiet coordinator, sensing changes in the internal environment and sending signals to maintain stability. It communicates through hormones that influence hunger, thirst, temperature. This dialogue ensures that homeostasis is not merely mechanical but responsive and adaptive, reflecting the body's needs as they shift through the day and night. Homeostasis is not perfection. It is resilience, the ability of the body to return to balance after gentle disturbances. After eating, blood sugar rises but soon settles. After movement, neurotransmitters shift but calm again. After sleep begins, hormones rearrange themselves to support healing and renewal. Each cycle brings the body back to its center. This, returning, is what keeps life steady and peaceful. As the biochemistry of nutrition helps maintain this equilibrium, it also supports the quiet transitions of daily life. Waking, moving, resting, thinking, and sleeping. The nutrients we absorb become part of this broader harmony, joining the molecular currents that flow inward and outward, supporting every cell that contributes to the sense of well-being. With this final part, the journey through the biochemistry of nutrition comes to a close. Carbohydrates, fats, and proteins digested, transformed, transported, and integrated become fuel, structure, signals, and balance. Their pathways reveal a landscape of calm motion, where every step unfolds in quiet coordination, and as these nutrients move through the body, they support the gentle homeostasis that keeps life steady